Okay, well, hi everyone. I'm Neil, founder and director of the Clinician Engineer Hub. We have our first virtual conference happening this year, which is really very exciting. And I have to say that over the next 3 days, what we've managed to do is probably quite an incredible feat. We have by far the best academics and best experts in industry across medicine and engineering coming to you completely free of charge. So this conference is starting at 1300 and it will go all the way up to 1800 UK GMT. Our first speaker, Professor Donald Ingber, is joining us from the United States, and it is an absolute pleasure and privilege to have him with us today. I'm sure anyone that knows anything about engineering knows Professor Ingber. He's literally done everything you can imagine in engineering, so completely very envious of all his feats. Not only that, he's contributed massively in terms of art, creativity, and design. So Professor Ingber is going to talk for the next 20 or 30 minutes, and if there's questions, we can then field that accordingly. So Professor Ingber, if you will.
Okay, I'm going to share screen here.
Awesome.
Let me put this on here. And can you see the Wyss Institute?
Yeah, we can see it perfectly. Yeah.
Fantastic. Okay, well, thank you so much. So I'm going to talk today about, I'm going to focus on human organs and chips from experimental models to clinical mimicry. At the top, you see the Wyss Institute logo. We've done a lot over the last 12 years. And just a background, I've been a professor at Harvard for, oh my God, 36 years. And but for the last 12, I've been the founding director of the Wyss Institute for Biologically Inspired Engineering. And the basic vision is that when we started really 15 years ago was that, you know, it's very clear that engineering has transformed medicine and the world actually for the better by taking engineering principles, trying to solve problems in biology, medicine, environment, et cetera. But we felt that we've uncovered enough about how nature builds, controls, and manufactures from the nanoscale up that we're actually at a point where we can now leverage biological principles to develop new engineering innovations. And that's what we call biologically inspired engineering as opposed to bioengineering. Now, when we started, we were lucky enough to get kickstarted with the largest single gift in Harvard's history, philanthropic gift of $125 million, which we've tripled since then. And what we were given that with the challenge of taking on high risk, high impact problems and challenges that, you know, conventional government agencies would not fund. We were not supposed to do incremental advance, they're supposed to do major leaps. And the biggest problem that I could see at the time was that the drug development model is broken. And, you know, and this is sort of well accepted across the board. This number of 2.6 billion of the cost of taking a drug from the bench to approval is from about 5 years ago now. It's probably more like $3 to $3.5 billion. And it's just a huge amount of money and time, and many reasons for this. One is that a lot of work is done with cells and culture that we know don't function anything like our bodies. You have to do animal studies to get approval required by regulatory agencies, and they often take years to complete. They are expensive. There are major ethical issues that I think you're more aware of in Europe than we are. And also the big problem is that 70, 80, 90% of the time in different areas, the results do not predict what we see when we go to clinical trials. And as a result, there's a lack of new drugs reaching patients, and there's been a search for better lab models that not only mimic cell function, but mimic tissue and organ level functions. So when we started the Institute, we had 5 major focus areas, and I led one myself that originally was called Biomimetic Microsystems, but really what we developed are what we call human organs on chips. And the idea was to engineer microchips containing living human cells that reconstitute organ level functions, not cell or tissue, but organ level functions to accelerate drug development, replace animal testing, and also advance personalized medicine. Now, why microchip manufacturing? Well, this is the manufacturing used to create computer microchips. It offers control of features at the same nanometer to micrometer scale that living cells and tissues live at. And it, commonly uses photolithographic etching, where you have a mask with a pattern in it, and the UV light will dissolve certain regions, not the others, and then let chemicals get to the photoresist, and you could etch patterns. And that's normally done in a clean room, very expensive, very slow. People wear those bunny suits. And around 1990, George Whitesides and I started collaborating, because George had developed an inexpensive way to make microchips, where you would etch with photolithography, let's say a pattern to make circular holes in a photoresist. Then you pour a liquid polymer, polydimethyl siloxane, a silicon rubber on top. It would solidify, and you now have a rubber stamp that amazingly would retain surface topography down to 60 to 90 nanometer resolution. And then you could stamp chemical inks. We originally used self-assembling monolayers at the nanoscale. Now we could just go directly with extracellular matrix, and we could surround that with non-adhesive regions like polyethylene glycol coated. And using that, we were able to engineer adhesive islands on the scale of individual cells. And in a series of science papers and Nature papers, we showed that you can control cell growth, apoptosis, differentiation, directional motility, stem cell switching, all by controlling that microenvironment. George went on to make what are called microfluidic systems. We have little hollow channels that join together to make bigger channels, inlets and outlets. And this is obviously you may know it has miniaturized instrumentation and diagnostics, but part of my work has been in vascular biology, and to me these were basically engineered microvascular networks. So we put this all together and created what we call a human breathing lung on a chip. And our first success was published in Science 10 years ago. The idea here is not to engineer a whole three-dimensional organ. If you're going to do that, just why not perfuse an organ? The idea is that we wanted to have sort of a window on molecular scale activity inside living cells, inside in a tissue and organ context, and be able to study that, visualize that, and analyze it in a controlled manner. And so what we've done is we created what I like to describe as living three-dimensional cross-sections through a major functional unit of a human organ. And so we started, as you see here, with the alveolus, or the air sac. And as I think you probably know, this is a relatively simple structure. It's sort of a bag of grapes, but it's hollow. If you go to the electron microscopic level, you have air. You have a single layer of alveolar epithelium, a porous, flexible extracellular matrix, the basement membrane. And on the opposite side, you have a capillary endothelial cell, and then you have blood. What it doesn't show, and of course, this is where you have gas exchange, anesthetic delivery, airborne drug delivery, smog particulates, metastases. So it's a very dynamic organ level site. And what I don't show is that, like all organs, it's an incredibly active structure from a mechanical perspective. Every time you breathe in and out, they stretch and relax. Blood is flowing, air is flowing, and we know from respiratory physiology that these physical motions are absolutely critical for lung development and maintenance of function throughout adult life. So if you're trying to distill down, like, what are the minimal features of an organ, which is what we tried to do, what makes an organ? Well, an organ are two or more types of tissues that come together, interface, and new functions emerge. And often one of them is a vascular tissue. So one thing we had to do is recreate this tissue-tissue interface. We believe that mechanical forces, and this is something I've pursued for 40 years, is that mechanical forces are important as chemicals and genes. So we wanted to integrate mechanical dynamic forces like dynamic flow and cyclic breathing motions. So I'm going to show you next is an animation of how this works. Top right is the chip. It's the size of a computer memory stick made out of that optically clear silicon rubber polymer. If you cut it in cross section, there are 3 channels, each less than 1 millimeter wide. The middle one is cut into top and bottom by a porous silicon membrane. It has engineered pores that are big enough for cell migration. We coat that with extracellular matrix. For the lung chip, we have lung alveolar cells on top from human capillary cells on the bottom from lung. We just recreated the alveolar capillary interface. The trick is we put cyclic suction through the side channels. Everything's flexible, and it literally stretches and relaxes to the same rate and degree as when we breathe in and out. We could then put air over the top to create an air-liquid interface on the epithelium. We could then have medium with or without immune cells, or even whole blood if we put endothelial cells on all four sides for short times. Now, if this were to work, imagine you should be able to mimic organ level response. So if you, in case of an infection with bacteria, you get a tissue-tissue signaling at the organ level with cytokines being produced that induce the endothelium all of a sudden to express adhesion receptors like ICAM. The white blood cells that were just flowing by adhere, roll, diapedese, and engulf. So now I'm going to show you imaging through the device. These are fresh white blood cells. I know that because we took them out of my postdoc, Dan, huh? They're labeled fluorescently. You cannot see the endothelium because it's not labeled, and the epithelium is on the other side of the screen. To begin with, quiescent endothelium, they just flow by. Now we add bacteria on the other side. There's that signaling response, and these cells are pulled out under flow, and that's important because the initial adhesion is shear stress dependent for immune cells. Now we could also do any imaging you could do in vitro. So this is high mag, it's one neutrophil. Right about here, it goes through two unlabeled endothelial cells, and then it finds the matrix-filled pore, wiggles its rear end out of focus. And now you're going to see it by phase contrast coming out the other side. Now I'm going to show you the white blood cells red colored because the bacteria are in GFP green, and you see them being engulfed. So you just watched the entire human inflammatory response in lung at high resolution in this little chip. Now, we did many studies where we showed that breathing motions actually are absolutely critical, for example, for absorption of smog, mimics of smog particulates, nanoparticulates. And we had a science paper because we confirmed that in animal models as well. But drug companies that we started to speak to said, well, this is great, but what we're really interested in are disease models, because that's where the animal models fall apart. And also drug toxicity models. So we hit two birds with one stone by developing a model of pulmonary edema by perfusing the cancer drug interleukin-2, it's an FDA-approved drug, through the vascular channel. It's normally given IV, and its dose-limiting toxicity is pulmonary vascular leakage, leading to pulmonary edema. And so what we found at time zero, if you look above with air, this is what the epithelium looks like. If you now perfuse this through the vascular channel over 2 to 3 days, you see a clear liquid filling the airspace, which completely fills it by day 4. We also saw by cross sections fibrin clots in the airspace, which is normally only seen in autopsy specimen. And this mimicked precisely the same time course and with the same dose that this toxicity has been seen in humans. Now, we took advantage of a clinical renal physiology trick by using fluorescent inulin to quantify the fluid shifts. And what was interesting is we confirmed in this top dark blue, you can see that the squares, you get a large amount of fluid shifting into the airspace. But what we found is if you stop the breathing motions, red squares, you actually would have never seen this. So this would have not been easily detected in a transwall, for example. Now, this did not mimic an in vivo response. This was a prediction. No one's ever seen this before, the idea that breathing motions could affect edema. And so we developed an ex vivo ventilation perfusion model in the mouse where you could keep the breathing going or stop it while having blood flow. And we confirmed exactly the same response in vivo. Breathing actually motions increased the drug toxicity. Now, as I mentioned, I've worked on mechanobiology for many years, and the fastest signaling response that we ever found was when we have endothelial cells on a matrix and we stretch it in 5 milliseconds, we activate calcium influx through an ion channel called TRPV4. And GlaxoSmithKline, actually, I knew had developed an inhibitor of TRPV4 that was in development. I heard that the program was not doing great, and I thought maybe they'd allow us to use the drug, which they did. They didn't fund the work. And we found that this inhibitor completely inhibited interleukin-2 induced pulmonary edema in a human lung chip. They then tested this in dogs and rabbits with cardiogenic pulmonary edema model, a back pressure model, and got exactly the same results. And we had back-to-back papers in Science Translation Medicine in 2012. And that drug is now in phase 2 clinical trials. And I really believe that this was part of helping the entire process move forward in a more rapid manner. I will also say that the reviewer on that paper said this should not be published. It's too simple a model. There are no immune cells in the chip. The other reviewer said, This is amazing. This is like synthetic biology at the cell, tissue, and organ level. And they could actually show you don't need immune cells to get pulmonary edema induced by interleukin-2. And this is something we've seen again and again, that there are many assumptions in medicine that we discover are wrong because normally there's no way to test them. But in these models, we can independently control every parameter, the cell types, whether there's breathing motions, whether the flow rates, whether we have growth factors added, and it basically, you can vary each one of those independently. Now, this one model, the lung alveolus chip, as we call it now, has demonstrated proof of principle for human disease model, drug toxicity model. We also, I'm not going to show you, but we have a paper where Janssen Pharmaceutical had a biologic therapeutic antibody for lupus, never saw toxicity in animal models, went to human clinical trials, multiple patients died due to pulmonary thrombosis. We can mimic that on our chips with blood. We have drug efficacy, I showed you, new therapeutic targets, and new drug discovery, as I mentioned. And this has won, numerous awards. I'm very proud it won the NC3Rs award in England, also won the International Design Award from the London Design Museum. It beat out a Google car and a Frank Gehry building. My wife is most proud that it's in the permanent collection of the Museum of Modern Art. I'm a permanent member, which means I get a permanent discount at the gift shop and the cafeteria, which makes all of us very happy. And it was named one of the top 10 emerging technologies by the World Economic Forum. Now, we've integrated immune cells, as I mentioned, but we've also integrated stromal cells, and you could add increasing complexity as you like. Now, these pharma companies said this is really interesting, but the diseases they were more interested in were asthma and COPD. So those are diseases of the small airway, not the alveolus. So we took this chip, we made it taller, 1 millimeter high. It's the radius of a bronchiole. Now we put primary human bronchiolar epithelial cells that normally form a mucociliated epithelium at an air-liquid interface. We grow them for 3 weeks on chip, only feeding through the vascular channel, like in our bodies. And at 3 weeks, they are a beautiful ciliated epithelium. They have directional cilia, move in a directional manner. This is an electron micrograph. If you put small fluorescent particles and you quantify mucociliary velocity, this is what you see. This is real-time imaging. This is the same exact velocity as in our lungs at this very moment. So we then made chips with cells from patients with chronic obstructive pulmonary disease. And what brings these patients to the ER is usually exacerbations by either bacterial infection, viral infection, or cigarette smoke. First amazing thing we found was that after 3 weeks on chip without any immune cells, we saw retention of the COPT phenotype and that there was decreased expression of TOLR3 and 4 receptors. This probably means it's epigenetic. The second thing is that when we mimicked viral infection, and I should say this is before we had approval for viruses, now we're doing influenza, coronaviruses, and much more, but we used poly-IC to mimic viral infection, and we used LPS endotoxin to mimic bacterial, and we found that we could see that LPS endotoxin exacerbates COPD chips by increased IL-8, but not healthy, and viral stimulus induces MCSF, which may in fact be a specific biomarker for viral exacerbation in the future. Now at the Wyss Institute, we could kind of do anything. So we built a cigarette smoking robot. This is a cigarette lighter from a car, a Gatling gun with real cigarettes. It mimics breathing parameters that you actually, the puff rate, the frequency, pauses, and the real smoke goes all the way into the airspace of the chip. This is not cigarette extract like it's usually done. When you do that, and I'm just going to show you 2 examples. What you can see at the left, measuring IL-8 as a sample cytokine. Normal chips from normal patients, no change in IL-8. COPD exacerbated, doubling in IL-8. Nowadays, we do multi-omics on these chips. So we do proteomics, metabolomics, glycomics, and transcriptomics. And at the right is transcriptomics. The 3 lanes at the far right are from 3 chips that are healthy chips exposed to cigarette smoke. The 9 at the left are from a publication, are healthy people who smoke cigarettes. What was impressed to us was that the top quarter to the bottom 3 quarters look pretty damn similar. That apparently did not impress the reviewers because they said, well, your chips are also similar and the patients are much more variable. And then we realized that we could actually do this type of clinical study better than a clinical study, in that we're doing matched comparative modeling of the same patient before and after a stimulus that we're interested in getting insight into mechanism. So all these changes are due to cigarette smoke, where in general, it's true here, but they also have different types of cigarettes they smoked and different working environments and different, histories and so forth. And that actually got into cell systems. And then we were asked to do a little mini review talking about new models, new ways of doing matched comparative models in humans in vitro. Okay, so we now had these two different organ chips, organ unit chips. And, I've worked in cancer since the 1970s. And in the cancer world, people used to inject tumors under the skin subcutaneously. In mice, they never really looked anything like in patients. And then people developed what they called orthotopic models in mice, where you inject a mammary tumor in the mammary fat pad or a prostate tumor into the prostate gland, and they looked a little bit more like in patients. So we had these human microenvironments. So we said, let's develop human orthotopic models. And we started with lung cancer. We used cells from a human non-small cell lung cancer, an adenocarcinoma form that clinically always emerges in the distant bronchiole, but almost always grows out in the alveoli in patients. We put these cells, we plated them the same time as the normal epithelium at a low ratio on the upper channel with endothelium on the bottom. And here you could see alveolus chip with cancer cells in green. They actually grow out and invade into the endothelium. But when we quantify, and then we did the same thing in an airway chip. And when we quantified the growth, what you see here is the gray circle is conventional culture medium on a conventional dish. They grow very rapidly. What you see in the white circle is the medium we use in our chips grown in a conventional dish. They don't grow at all. And then the black squares are what we see on chip in the airway chip. You can see there's no growth for 10 days, and then they slowly grow. All of this growth is due to the microenvironment of the chip. It has the same medium on a plastic dish. They do not grow. Basically the endothelium, the epithelium, and the same medium is used for both, and the air-liquid interface. When we put the cells in the alveolus chip, which is where they grow in vivo, they grow extremely rapidly on chip. And so we mimic that microenvironmental difference. But what was interesting is when we looked at plus or minus breathing in the alveolus chip, we saw that breathing suppressed growth by 50% on chip. We also found that we could easily quantify invasion because we could watch the green GFP tumor cells migrating through the pores. And when we looked at breathing, that also inhibited invasion by 50%. Now, we went further and we looked at response to ruxolitinib, which is a frontline tyrosine kinase inhibitor, and we found that breathing regulates the therapeutic response as well. So for example, with no breathing, the tumor is growing rapidly and you inhibit it potently. With breathing, the tumor grows slowly, but it keeps growing right through slowly, even in the presence of this potent drug. And we could show this is regulated at the level of EGFR phosphorylation. Now, this is interesting clinically because, imagine one of us has a tumor just beginning in the distant bronchiole, we're breathing. It now moves into the alveolus and we're breathing, and it's slowly growing over 20 years. And then all of a sudden it fills an alveolus that will rigidify it. When it gets rigidified, this is saying there would be a positive feedback loop saying it would grow more rapidly, which would then fill the next alveolus and so on. And this would just grow rampantly. You then treat it with drug in patients to get a rapid shrinkage, send the patient home, and then basically they have residual disease that grows slowly and they come back. And this may be a way to begin to study this in vitro. We went on to develop many other chips. So for example, we developed the human intestine chip. We started with human CACO2 intestinal epithelial cells, which normally grow as flat cells on trans walls used by pharma for barrier function. Normally in vivo, they're columnar and form villi. Put them on the chip with flow and peristaltic-like motions for 5 days, and you get villi, and they differentiate into all 4 lineages of small intestine and put out mucus. More recently, we've moved to primary intestine chips using cells from patient-derived organoids. We've done duodenum, jejunum, ileum, and colon. This is a duodenal chip looking from above and from the side, and this is Ki67 for proliferative cells that are at the base, and you can see you get beautiful villus formation on these chips. When you do transcriptomic analysis of the duodenum for which we isolated organoids, the organoids at the left, and then our chip across the whole transcriptome, we actually could see that our chips are closer to in vivo than organoids. And organoids are wonderful, but you can't get access to the lumen, you can't look at transport absorption, and you can't look at mucus, for example. Our colon chip we published recently, not only does it differentiate absolutely beautifully, and this is a histological section through the chip with villi at the top, basal nuclei, goblet cells, etc., but you could turn the chip on the side and in real time visualize mucus production using DIC optics. And this is same culture over 14 days. And we could also show for the first time that we reconstitute the thickness and the bilayer structure of human colon mucus layer in vitro. But the most important thing I think we've done is that, and the biggest paradigm shift in medicine, I think in the last 15 years, is the microbiome. And almost everyone studies microbiome at the metagenomic level, because if you culture them with human cells in a dish, we call it contamination, they die. But with the chip, we developed a way where we could develop a homeostatic environment and co-culture them for extended time. So first, we created a hypoxia gradient such that we have an aerated medium going through the endothelium line channel, but we have nitrogen gas around it in the other channel, and we have oxygen centers integrated into the chips, which we could read out levels, and we could show that we could sustain oxygen levels down to 0.3%, which is, you need below 0.5 to get obligate anaerobes to survive. And as you know, the microbiome is a mixture of aerobic and anaerobic. So you basically have 0.3% up in the lumen. You have enough oxygen in the endothelium and the epithelium to survive, just like in vivo. And now we've been able to culture complex microbiome on these chips taken from stool specimens and show we get aerobic and anaerobic bacterial growth. And interestingly, with this complex microbiome, we get better barrier function than without microbiome. And this is the lower level, the apparent permeability, the better the barrier. And this just gives you a feel at the right. This is a video where we took from a neonatal intensive care unit at Children's Hospital. We had a baby with stool specimen. All these little speckles between these villi, that's the density of bacteria we have on this chip. And we could grow them for at least 5 days, and the cultures are perfectly happy. And this is the level of complexity of microbiome comparing stool to what we get on chip, very similar. We have over 200 different types of bacterial strains or operational taxonomic units from 11 different genera. So for the first time, we could study complex microbiome from patients in contact with living human intestinal epithelium and their mucus layer. We've developed other chips, for example, kidney glomerulus chip. Here we had to develop a technology to develop kidney podocytes because there's no cultured podocyte that's differentiated. We developed our own IPS strategy where we gave it embryonic matrix and also the physical forces of the chip, and you get beautiful podocytes and processes. Now we put the kidney podocytes on one side, glomerular endothelium from human on the other, and we give it 1 beat per second of stretch, 10% strain, because I lectured at the kidney meetings and the person before me had done imaging of the glomerulus in animals, and you get every beat of the heart once a second, you get about 10% strain. And only when we give it that mechanical stimulus do you see foot processes going through the pores, touching the basement membrane of the endothelium, much like in vivo. And only with the 10% strain do we reconstitute urinary clearance rates on chip that mimic that in vivo, measuring inulin versus albumin retention. We've also made a blood-brain barrier chip with another human IPS-derived brain microvascular endothelium that we have on the lower channel. On the top, we have human astrocytes and pericytes. You get a beautiful endothelium with tight junctions and transporter expression. And for the first time, we're able to reconstitute barrier function like you see in vivo. The little dotted line down here is the transepithelial electrical resistance in ohm centimeter squared of what we and others have got in the past, which is around 60 to 100. We're now up to the 2,000 to 8,000 level, and we could maintain that for a week in culture. We also could reconstitute brain shuttling capabilities that Genentech has demonstrated with transferrin receptor antibodies. So we're now using this to discover new brain shuttles. Okay, so pulling to the end. So the ultimate goal, though, is really to recapitulate human pathophysiology and predict human responses to drugs using clinically relevant dose exposures. Almost everything we all do in vitro, we bathe cells in drugs. You know, example is 5-fluorouracil with bone marrow cells in culture. You can show toxicity, but you make them sit in it for 5 days, and you're using a concentration that doesn't scale with a 2-day infusion in humans. So recently we published a human bone marrow chip where we literally, all we do is we take CD34 cells from bone marrow or blood, we combine them with bone marrow derived stromal progenitor cells and put them in a fibrin gel, put that in one channel, we put endothelium on the other, we perfuse through the endothelium, and we get blood development over a month that looks just like in vivo in terms of neutrophil maturation, erythroid differentiation by FACS sorting. This is a Guim sustain with all different cell types. We could show that we maintain progenitor cells and we get much higher levels than you would in classic suspension culture. We're funded by the FDA on this actually, and part of it is to look at radiation countermeasure development, and radiation toxicity models on animals don't usually show the same dose sensitivity as in humans. We could show, and each color here is a different patient chip, so show really nice patient-to-patient reproducibility, and we get in the 1 to 2 gray range of exposure toxicities, which really matches very nicely to what you see in patients. And you see the progenitor cells at left are more sensitive than the mature, as you'd expect. We then went and did the 5-fluorouracil study, and what you see here that is highlighted are the levels of drug in patient plasma, which is about 4 micromolar with a 2-day infusion. And at that dose, with our chips at the bottom, we see nice toxicity as seen in patients, where you do not see this in the same chip without flow, just static or in suspension culture. But what was really interesting is that AstraZeneca, the pharmaceutical company, came to us, and they have a drug in clinical trials, and they had seen very bizarre regimen-specific toxicity, where it's a cancer drug and they give the same dose at 2 hours or 48. At 2 hours, they get neutropenia and anemia. At 48, they get only neutropenia. They had the pharmacokinetics measured by mass spec in patient plasma in gray. We could mimic that precisely on chip because we control flow. When we mimic their PK profile, we precisely replicate the toxicity of anemia and neutropenia at 2 hours, but not at 48. And this cannot be done in suspension culture. We could also model recovery, which couldn't be done in animals. And we then went one step further. We made chips with cells from patients with a rare genetic disorder called Shwachman-Diamond syndrome. And they have basically defects in bone marrow development. And even looking at the low magnification, you could see chips made with their cells have much less development of cellularity. When we quantified it, we could see they have less neutrophils, less erythroid cells, less CD34 cells, and they were less mature in terms of neutrophil maturity in particular. What was really interesting is that we found, we observed abnormal neutrophil maturation phenotype on chip that has never been noticed before, which was abnormal low CD13 to CD16 ratio by FACS sorting. And when we went back to the clinical samples of patients, we found that the 2 patients we had analyzed were in a subset of patients that had this phenotype. So not all patients had it, about 50% had it, but we were able to uncover some new insight into mechanism of pathophysiology using these chips. And I think a real excitement here is with rare genetic disorders. It's very hard to do clinical trials to pull a lot of patients at one site, but you might be able to pull CD34 cells and do trials on chips, and then plan a bigger trial later on. Final example on chips is we had pharmaceutical companies, 2 different companies come to us and say, could you do rat, dog, and human liver chips? And the reason is that every pharma biotech company has to do preclinical hepatotoxicity studies for the FDA or European agency and they almost always get conflicting results. So we did that, and we literally made liver chips that have human, dog, or rat primary hepatocytes, species-specific liver sinusoidal endothelial cells, species-specific Kupffer cells, and species-specific stellate cells. And we reconstituted this all, and with that, long story short, we could show that these faithfully recapitulate species-specific hepatocellular injury, steatosis, cholestasis, and fibrosis toxicities with multiple compounds from two different major pharma, Janssen and AstraZeneca, and that was published, I think, in 2019. So this, I think, really can change the paradigm of how drug development is done. So we've now developed probably 20 different organ chips. But when I first presented this, I had a review in 2012 that because we have a vascular channel lined by endothelium, that we might be able to create an integrated human body on chips by connecting flow from one vascular channel to the other. So you can imagine putting an oral drug to the lumen of the gut chip, watch it be absorbed, metabolized by the liver chip, peed out by the kidney chip. Do you have heart toxicity? What does it do to bone marrow? Or an aerosol drug in the lung chip? Long story short, we have done this. We published this year. First of all, we don't connect with tubes. We use a robotic sampler that can link to pharma and drugs, but then it also links chip to chip by drop to drop. So we could take a drop, go from chip A to chip B, take a next drop and do mass spec for drug levels, for example. And the next chip can go to B to C and so forth. And in this study, we were able to show that this automated instrument cultures them, allows imaging, controls flow. And we did gut, liver, kidney, heart, lung, blood-brain barrier, brain, skin, bone marrow, and pancreas for a month. We were able to keep them alive and functional. But the real goal was to use this to predict human drug pharmacokinetic and pharmacodynamic parameters, PKPD, with the goal of maybe be able to shortcut clinical trials design in the future. And to do this, we simplified, we made a first pass model of a gut, liver, kidney chip. We had a proximal tubule chip, and we mathematically approximated for the glomerular filtration rate. We integrated an arteriovenous reservoir to have mixing so that you could take samples from it like a venous blood sample. And the goal is really to pull out by carrying out measurements on chip and then using computational modeling, basically physiology-based PKPD modeling to pull out parameters like Cmax and T1/2, maximum drug concentration and half time to get to max. And we have done this also, and we had 3 papers in the same issue of Nature Biomedical Engineering in January. In this one, we had a gut chip, liver chip, kidney chip, AV reservoir. We put the drug nicotine as an oral drug, like chewing tobacco, into the gut chip, watched it be absorbed, measured metabolism, clearance, and took values in the AV reservoir. And then separately, we did intravenous cisplatin. Now we have a bone marrow chip to look at pharmacodynamic effects, but we have liver, kidney, and a reservoir. And what we see at the right are the dotted lines are our computational models done with a company called CFDRC that basically scales from our chips to human organs based on blood volume and flow rates and mass and so forth. And the dots with the error bars are from past publications in humans, and we precisely mimic the PK for both of these drugs using the same computational model. It's demonstrating this is actually feasible at this point. So to end, I think, what we can say is that our 2-channel organ chips, and I say 2-channel because a lot of people use the term organ chips and all they do is have cells under flow, but we really feel like you need a tissue-tissue interface to have an organ chip rather than a tissue chip. That they faithfully recapitulate human pathophysiology. They mimic human response to drugs and radiations using clinically relevant dose exposures. They could quantitatively predict human drug PK. And we believe that they're now ready to be integrated into the drug development pipeline. And they, in fact, are being used around the world by many different companies and academic labs at this point. I just had a paper come out 2 weeks ago, and this is literally the title. Is it time for reviewer 3 to request human organ chip experiments instead of animal validation studies? I invite you to look at that. It sort of sums it up. And the way I usually like to end is to say that the way big pharma companies now do drug development is they spend tens or hundreds of millions of dollars to develop it and do huge clinical trials, which almost always fail. And then they'll have statisticians do number crunching for a year or so and try to see, is there a genetic subgroup that might have responded better? And if so, they'll do a small targeted trial. And if they're lucky, they'll get approved for a narrow application. With organ chips, iPS cells, organoids, you can imagine taking, let's say, 50 women who have asthma and COPD who are ultra sensitive to cigarette smoke, make chips from them, design drugs for them, and have them be your small clinical trial. I think that kind of approach could shorten the development time, decrease the cost, and increase the likelihood of success, and really transform how we do drug development. The other side, of course, is individualized organ chips for personalized medicine, where you might have your intestine chip if you have IBD, where you're trying to see which drugs suppress IBD while minimizing toxicity in your liver. And this actually is being done. There's hospitals like the Cedars-Sinai Medical Center in Beverly Hills, which actually is iPS cell programs they've had for years, and they are integrating iPS into chips, and hopefully in the future we'll be using it for drug selection on patients, or as I said earlier, maybe for clinical trials with rare genetic disorders. That's something that could be pursued as well. So to end, I cannot do this without incredible range of know-how, which is what we do at the Wyss Institute. Andre Prequis is the person at CFDRC who does the computational modeling, and Kit Parker helped develop some of the chips that I did not describe that were integrated in the human body on chips. I have to tell you at the end that as a Harvard faculty, I have to fully disclose that you can't believe anything I said, because I hold equity and chair the scientific advisory board of a company called Emulate that I founded, that is now selling these chips and automated instruments to run them around the world. And with that, if you want to learn more, please come to our website. We've won 3 Webbies, which is the Academy Award of the Internet for websites, as well as other awards. I think you might really enjoy it. So with that, I'm happy to answer questions.
Thank you, sir. You definitely, Prof. Ingber, put a lot of pressure on the other speakers for the rest of the day to match that level of expertise. I'll probably start. So I think obviously lung cancer you spend a lot of time on, and as someone that does internal medicine and gastroenterology, I see a lot of probably chest infections as opposed to lung cancer. Do you think there's any potential for the organ-on-chip platforms for people with nasty pneumonias, that even though we throw antibiotics at them, for some reason it just doesn't work? They're either resistant or they keep getting repeated chest infections.
We've been funded by the Cystic Fibrosis Foundation. We've reconstituted cystic fibrosis on chip. We have Pseudomonas growing in chip. We have been funded for 3 years to develop chips to model pandemic virus infections.
Sure.
So we've modeled influenza on chip. We can model the host response, the different virulence of different strains, the immune cell recruitment, cytokine storms. We've done viral evolution on chips, where you can see spontaneous evolution of resistance to drugs. We've discovered new mutations. And then with COVID-19 in February, we developed pseudotype viruses for SARS-CoV-2. And we have drugs that we've discovered that work in animals, that both for prevention of animal-to-animal transmission and therapeutically. And I have a huge DARPA grant. We've screened through 500 drugs using various models, not all on organ chips, but the idea is to use organ chips as sort of another validation model. So the answer is yes, we do a lot of infection. In intestine, we have models from Gates Foundation on environmental enteric dysfunction. With microbiome, we could reconstitute phenotype. We've shown viral infection with Coxsackie viruses on chips. So yeah, it's great for infection models.
Awesome. And I'll also add that obviously very interested in the inflammatory bowel disease side of things. Yeah. As you know, we often throw biologics at patients and they either work for a year or then they get a loss of response. So it's very interesting to see how this technology will move the field forward.
So just 2 things for you that are very interesting. I probably should have described. We had an early paper on IBD model with the intestine chip where we showed that you can grow microbiome on the chip and they're in balance, but if you don't have peristaltic motions, you basically get small, intestinal overgrowth of bacteria. So, and we still had flow. So the idea that you get ileus in patients when you don't have flow is wrong. It's when you don't have peristalsis. And then the other side is you can collect cytokines out of the vascular channel. So when we modeled IBD, we found a combination of 4 cytokines, and I'm forgetting what they are, but we also could measure their levels, and then we could add them back without the stimulus for IBD, the inflammatory cells and bacteria. And when we add those 4 cytokines, only with the combination of 4 at that concentration do you get injury and barrier disruption. So you can discover like what combination lock of cytokines cause injury.
Absolutely.
Yeah.
Definitely.
So those are the types of things I should have described that you can do on chips you can't do any other way.
Sure. Do you have time for any more questions? The
Yeah,
audience
sure.
are desperate to ask you things. So, got a question from Rishi, who says, for a personalized tumor on chip model, is there a limit to the size of an explant you could integrate to be able to assess the behavior of specific tumors?
Yeah, so I mean, we're dealing with very small channels right now. So, and we're dealing with sort of cell level. However, and I should say I have a Cancer UK Grand Challenge grant where we're modeling esophageal cancers and colon cancers. And we've got some nice results with esophageal cancers with metaplasia, dysplasia, and so forth. But the channels that we use are like 1 millimeter high. We do have a chip that has an open top that is much bigger that we've done for skin that could be used for a larger, many millimeter, almost a centimeter size, but that would be, but then you can't visualize what's going on. So we have been focusing on smaller size.
Okay, awesome. Questions come from Sam Venin, who says there are more and more in silico computational models being used for drug development, notably to test toxicity. According to yourself, what are the comparative advantages, weaknesses of such models versus organ on a chip model for drug development? Is there a path where both could be used in combination?
Oh, absolutely, both would be used in combination. I think the PK model shows that precisely. I think the problem is, and I've done a lot of computational modeling over my career, I think the key of computational biology is that it's iteratively linked with data. If you just try to pull it, I mean, with experimental data and experimental testing and validation, and you kind of auger in on a solution rather than think you're going to just use big data from the web. We use artificial intelligence machine learning to do drug repurposing based on transcriptomic data from patients, but also then from chips.
Mm-hmm.
I think, I don't think in isolation just from data, big data, you're going to really do anything useful, but I think you will have to integrate computational models with human-based data sets, and that could be from patients or it could be from chips. The thing with patients is you could only get the complexity result. With chips, you could begin to sift out like which part of this is due to which tissue types, organ types, what is due to, you could get more linkage to cause and effect. But they both will be used together, absolutely.
Great. Do you have time for some more? Or
Yeah. No, I got a few minutes, sure.
Okay, so no name provided for this one, but have you tried or thought about trying lymph node chips and testing response to vaccines?
Yeah, we've done that. It's been for 2 years or year and a half in bioarchive. So just search my name and lymph, and lymph. Yeah, we take blood cells at high density in a gel and we put them in one channel, and amazingly we get germinal center formation, antibody class switching. We can put commercial Fluzone, which is influenza vaccine, in the chip, and we get the entire response, IgG production and even cytokine response similar to humans, because we had lucky enough to have samples from humans. We're resubmitting that to a major journal now, but you could look in bioarchives. And we have funding from both pharma and Gates to look at adjuvant testing right now of vaccines using that.
Fantastic. Time for one more, I think.
Okay.
Dr. Araber has asked, how does genomics and epigenetics relate to microbiome chips?
Well, that's a pretty high level question, but we have not done enough in epigenetics, but there's no doubt, like I mentioned with COPD is thought to be an inflammatory disease clinically, which it is.
Yeah.
Yet we take these cells out, we buy them from a company who isolate them, culture them, we culture them, then we put them on chips, then we differentiate, and they still retain the phenotype. That to me is amazing. It has to be epigenetic. So I think we need to look into that more and more with chips, but when we can retain phenotype on chip that involves a complex environment in vivo, that's telling you a lot. Cystic fibrosis is, another example. In terms of genomics, it's basically you could use the same tools that you would normally use in patients, but now you could do it in a much more controlled way. As I was saying, you could start, like we usually use a synthetic biology approach. We'll start simple. We'll just put the epithelium on. Do we mimic a complex phenotype in vivo, in human? That's our benchmark. Not, do I see the effect of this drug, or do I see the effect of this chemical, or does it look like a cell? We do in vivo mimicry. And so if no, something's missing, then we'll put the endothelium, or we'll put the stroma, or the immune cells. If yes, maybe it's simpler than we all thought. Maybe that is a feature of that one cell type or those two cell types. And so you can do genomics in those contexts where you have simpler systems, and maybe counterpoint that with what you see in patients. Like, with COVID now, we are getting data. We have a big grant where I don't have a BSL-3, but my collaborators do. So we have SARS-CoV-2, original SARS-CoV, we have MERS, then we have influenza, and we have this in mouse, hamsters, we have human autopsy specimens, and then we have, models with pseudotype virus. Now you can counterpoint all these things from a genomic perspective, and you begin to get insight into what is, late level response, initial response, which cells triggers what. And I think it's this synthetic biology idea that people miss. It's like, oh, they're already in vitro models. This is the usual review of our papers, right? And there's a model of this, and there's a model of that, and there's a model of this. But in one thing, you can model all facets of it, and it's human, and you have the inflammatory response, and it's underflow, and you could collect cytokines. The reviewers usually don't see that, but it's that, and being able to counterpoint it with real clinical data. I think that's really what allows you to get new insights.
Yeah, and I think it's important, actually, when you touched upon your talk, you mentioned a lot of the battles that you face, all the data that you find. You're trying to impress the reviewers, which obviously often put restrictions on barriers. But yeah, great, fantastic. Well, thanks very much for your time, Prof. Ingber. It's really, much appreciated. And
My
I
pleasure.
realize it's very early over there. So thank you once again, sir. Thank you.
Okay, thank you. Bye-bye. Pleasure.
All the best. Take care. Bye. Okay, everyone. So we're going to continue with the conference. Our next speaker is due at 2 p.m. So there's going to be a slight conference break, and then we'll kick off. So stretch your legs, have something to eat, drink, whatever you like to do. It's important not to sit down for the whole time. We'll be back soon. So our second speaker is Prof. Elisa Konofagou, direct from New York City. We were just doing some pre-prep in the background, so apologies for that, but this is a live event, so sometimes it doesn't always go according to time. So Prof. Elisa is an absolute expert from New York in terms of tissue mechanics and also drug delivery, so really honored for her to give the time today. Again, speakers from America are having to wake up slightly early, but I'll let her crack on because I know how busy she is, and I used to live in New York, so I know how busy New Yorkers are. Prof. Elisa.
Thank you, Neil. It is a pleasure to be here, and it is the start of the day here, so pardon the delay. So I want to share with you a couple of things that we've been working on at Columbia over the years, and hopefully have some time after that for a few questions. I'm going to first preface it by where we are. So Neil probably recognizes this. It's in the Upper Manhattan area, and this is on the left, the Law Library, where we have the president of the university and all the offices of the provost. And on the right, we have the engineering school, and it's been around for a century and a half now. This is where we have the biomedical engineering department. And we also have a really strong affiliation with the medical center, which is a few blocks up further to the north. So just to orient you, this is the Bronx here, Manhattan. On the left is New Jersey, and this is the George Washington Bridge that links Manhattan to Jersey. So, and you can see how the density of these buildings on top of one another. This happened over the past, since the early 19th century, in fact. And the Vanderbilt Clinic building here is where part of our labs of biomedical engineering are, and the neighboring Presbyterian Hospital is also where a lot of the, half of our faculty biomedical engineering labs are. So, all right, I'm going to talk today mainly about ultrasound. I have just this schematic here to show you. There is the acoustic wave that we use. You can see this propagation from the top of a source that's oscillating, and it has compressive and refractionary or tensile forces on the medium that it propagates in. And we use it in my lab with two different ways. One is to perform imaging, and the main part of imaging is on cardiovascular. But we also, I'm going to show you some of the breast cancer work that we're doing. And then also therapeutics, which is less known, but it's gaining very fast acceptance to the clinic due to its non-invasive properties. So therapeutic ultrasound is where you can use ultrasound waves to either mechanically move tissues or burn them, or both. And we do that in the cancer realm, but also in the brain. So first, the cardiovascular imaging aspects, we do both anatomical, that's provided by classical conventional ultrasound, but we also do functional imaging. And the main idea is to see the elasticity of the underlying tissues. So on the heart, we look at the actual mechanical properties, both as it pertains to the underlying scarring as a result of cardiovascular disease or infarction, and then the electrical properties. And in the vessels, we continue looking at the function as the stiffening happens anywhere from natural aging or due to atherosclerosis. So first things first, the cardiac realm is where we have a lot of the clinical studies going on. We start, we call the technique myocardial elastography because it maps the elasticity around the myocardium. Of course, the most important part of the heart, the chamber of the left ventricle, is the one that's the most muscular, but also the one that could fail, leading to heart failure and even organ failure. So where you see red is actually what you want in a normal heart, which is thickening during systole. Where you see blue is actually what you don't want, is the opposite thinning that happens as a result of obstruction of coronary arteries. In this case, the bottom left is right coronary artery, and then the bottom right is where you have two different stenosis, one on the LCX and the other one on also on the right coronary artery. So we're mapping that at early stages, and we're trying to see whether we can detect from normal to ischemia or scarring. We're able to do that based on the strain across the entire heart. And we see that we can do this in multiple territories. And we can differentiate between normal ischemia or scar, normal versus ischemia, and normal and scar only. And then when it comes to territories, we also can do that across different territories. So we have these 3 coronary territories in the left ventricle. And this is a preliminary results that we had published a couple of years ago. We're continuing that with hundreds of patients, hoping to see also how small of an occlusion we can detect. The second part I want to share with you today is on electromechanical activity. So something less intuitive is that we can look at electrical function with ultrasound. So obviously the most typical one is EKG that you see up here, P wave, QRS, and then followed by T wave. But what happens in the heart is that as you have the electrical signal propagating the Purkinje fiber network, the Purkinje fibers innervate the underlying muscles, and the muscles are actually contracting. So the idea here, something that's known at the cellular level, is to translate that to the tissue level and see how the action potential propagates electrically, can also propagate mechanically. You can see this red activation of the septum, the wall that separates the 2 ventricles, followed by the blue wave, the mechanical activation also of the same area. So because something that we have in ultrasound is that we can get really fast images, and over the course of the past decade, it has boomed with thousands of frames a second, even in a normal heart, and even in a heart that's actually a large organ that you need large depths. So what I'm going to show you is on the order of 1,000 frames a second, actually 2,000 frames a second, of course, slow down. And you see all the 4 chambers in the heart, like you usually have in what's called the 4-chamber view in ultrasound, with a perpendicular 2-chamber view. So right atrium, right ventricle, left ventricle, and we have put the Purkinje fiber network on top, and you're going to see in blue the activation. So this is in a normal subject, and obviously the activation starts from the sinus node in blue, followed by the left atrium, and then the 2 ventricles contracting in sync. So we can see a propagation of this blue wave. And in fact, if you make activation maps or isochrones showing the activation time, so in here in red we're showing early activation and blue is late. And we map this across multiple views of the heart. So you can do an interpolation scheme and link through EKG gating these multiple views. In fact, here we had, I think, 4 views. And we're able to map the entire chamber in 3D. And you see early activation in the preventricular contraction cycle. And in the same subject, the next beat, the next cardiac cycle, the patient comes out of the arrhythmia and has a normal sinus rhythm cycle. And we can actually see that also and map that as well. So through a mechanical, very fast mapping technique, we're able to see electrical activation. And in fact, we partnered with clinicians at Columbia, cardiologists who look at these traces from the 12-lead EKG and try to map, reconstruct basically where the actual arrhythmia may be happening. Here I'm showing you 2 atrial arrhythmias, atrial tachycardia and atrial flutter. And we were able, with the same technique, to say that you did indeed have an atrial tachycardia, but where in the atrium. In this case, we were able to pinpoint it on what's called the roof of the atria, in this case, the right atrium. So the electrophysiologists were really happy with this, because you could actually go into the ablation suites knowing where you're going to start ablating from, something that's not happening right now. And then the atrial flutter, we could see atrial flutter is actually kind of an infinite loop where the actual potential loops around the actual opening of the chamber, trying to activate the chamber, but not necessarily effectively. And you can see it starts from the septum, the atrial septum, and it loops around. And we can see that also in this particular flutter. In the ventricular arrhythmias, I already showed you the case on the right. We also have what's called the Wolff-Parkinson-White syndrome. That's a congenital arrhythmia. Typically, the atria and the ventricles are abnormally linked through an accessory pathway. So part of the ventricle activates together with the atria. So the electrophysiologist would like to know where to ablate, and this is a very similar way of showing you here that the source of the arrhythmia is close to the left ventricular base on the lateral side. And then when we compared with the actual 12-lead EKG, which of course is the gold standard, we wanted to do as well. And in fact, one of the serendipitous engineering research studies that happens very rarely, I have to acknowledge, but we did better than the gold standard. And in this case, we actually showed that electromechanical wave imaging has an accuracy of 96%, which is higher than 71%. We had 5 clinicians rate those arrhythmias with 2 different algorithms that they're familiar with. So we published this earlier this year. Now I'm going to slowly go to the vascular realm. As I said, we're doing functional imaging there as well. Very similar intrinsic wave. So that wave that emanates mechanically from the heart through electrical activation goes to the rest of the circulation through the blood volume that gets pumped out of the heart. And we all have felt our pulse on the carotid and our pulse on the wrist. Now we can map it. So here's what the actual carotid looks like on a regular beam mode. You may be able to see the movie, I don't know. Yeah, here it is. So you have a distension of the actual carotid. And then we can actually see when we actually map the motion on the actual wall. Hopefully you can see that. A very fast moving red wave, right? So this comes from the heart. So the heart is electrically activating, mechanically also having this wave, but then this is a hemodynamic wave that's pumped through the carotid in this case. And that speed contains information, of course, on the underlying elasticity. We actually have compliance that we're measuring. We can measure it across the entire segment and give it one number, such as in this case, we had a particular number for the carotid segment that I showed you, or you can split it into very small segments in millimeters and do a piecewise mapping. So this is what we started doing when we wanted to see how elastic a plaque is, because as you know, in atherosclerotic plaque, you have a local abnormality, and the idea here is, and not a new idea, but very difficult to map that, is whether you can predict how brittle and how soft, for example, the plaque would be. So normal carotid is blue. So here, that means uniform compliance and relatively high. What we saw also in plaques is that we can separate according to whether you have high compliance in normals or severely calcified, which we can all understand is very stiff. So that actually is also a stable plaque. And so physicians don't have to necessarily operate on that, but it's very difficult to tell from a regular echo whether this is a calcified plaque. And then, of course, lipid versus moderately calcified, the lipid one being the more precarious and most likely the one that you have to perform endarterectomy on to avoid stroke events. So we continue that study too, but there's another element that we ignored in the heart, but we don't ignore in the vessels, which is basically the underlying flow. So how the underlying flow affects the elasticity of the vessel and vice versa. And here is the normal, again, carotid, the left ventricular bulb. We have put these fiducial markers, and through two-dimensional tracking, we're able to visualize this fiducial marker motion. And you can see that there's a vortex here forming at the bulb. And in fact, this region here is the most prone to atherosclerotic plaque formation. And then we have mechanical models that are fluid-solid interaction on finite element frameworks. This is an FEBio that was built through a collaboration from Columbia Mechanical Engineering and I believe Utrecht in the Netherlands. And we tried to see how well we can map both the wave on the wall that you see here and the wave inside the lumen. And the two are, of course, sorry about that. The two, of course, are linked, and we have both the flow in vector Doppler, as we call it, because it's angle independent and two-dimensional vector Doppler. And we're able to see this in 50% stenosis, where you have pretty uniform flow overall and a slight vortex here of formation. And then a 70% stenosis, we're able to see that we have much higher vortex formation, especially post-stenotically, that we can also visualize with the actual fiducial marker flow alone. So that's affecting both the pulse wave, as we call it, for the compliance estimation, but also the flow itself. Now I want to transition to the second part of the talk. I know I have a couple of minutes, 10 minutes or so, for what we can do with therapeutics. In this particular case, for cancer, we collaborate with the Cancer Center in Colombia, and we're able to perform both imaging and therapy at the same time. I'm showing you here a face-on transducer assembly. So you can see the phased array that is used typically in ultrasound imaging, and that's something that we also used in the previous examples. But then we have it concentric with a larger array here that operates at higher power. It doesn't perform imaging. It has a particular geometric focus that I'm going to show you in a few slides, where you can actually burn or mechanically move tissues. So we do everything that I just explained to you at the same time, trying to both visualize the tumor here and mechanically probe it, and at the same time monitor ablation. So these are some of the examples that we did in the ex vivo breast. You can see the gross pathology on the left. We have the two-dimensional harmonic motion imaging. So this is where we map the underlying relative stiffness. So what you have in white is soft, and what you have in low intensity pixels is actually harder. And we can render this in 3D. And then we can look at that before and after ablation. So ablation typically is thermally ablating, means that you have to surpass 50 degrees C. So you can see that the lesion here, the thermal lesion, was larger and harder than the underlying tumor. This is an invasive ductal carcinoma. And the same thing for the fibroadenoma benign tumor. And of course, you can also do this in normal tissues. So we have moved in patients in vivo, and this is what our setup looks like. We have both systems for the therapeutic and imager. This is a robotic arm, sits on the breast of the patient that is supine, and these are some of the parameters that we use. And then we use the regular ultrasound B mode to visualize where the tumor is, and then our harmonic motion imaging, which we oscillate the tumor locally. And blue is low displacement, so higher stiffness. Red is high displacement, or low stiffness, so softer. And what you see on the leftmost panel is in vivo, so in the patient. So you see a darker region here, blue region that overlaps with where the tumor was. And this is ex vivo, so after surgery, we can actually also visualize the underlying tumor. And of course, in the region where the breast is normal, we have much softer regions. That correlated very nicely with pathology. And we were able, with this technique, to separate malignancy from surrounding tissue, the same thing with benign tumor. But most importantly, the benign tumors were shown to be having higher displacement or softer than the malignant tumors. And we also saw a difference between ex vivo and in vivo. So post-surgically, the tumors, the surrounding tissues seem to be softening. And it could be that they also have nonlinear properties. When they are constrained within the breast, you have higher stiffness, and then after surgery, after removing the actual tissue, it becomes less constrained and therefore apparently softer. We also have work going on in pancreatic cancer that we published also over the years. This is the normal pancreas, which is red, again is soft here, fibrotic, rapidly becoming stiffer, and the pancreatic tumor, of course, even harder than the underlying surrounding tissue. And something that we've seen here is that while tumor progresses, we have a very much higher stiffness, and this is actually inverting now the color map to red, because cancer oncologists are actually used to seeing red for tumor. And you can see also statistically that in that mouse tumor model, we were able to separate clearly the tumor with the displacement, and also that correlated very nicely with this precorious red density here, which stands for collagen. So as the collagen multiplies in the tumor, you have higher stiffening. And that's something that we've seen both in the tumor progression, but also as a result of chemotherapy. So chemotherapy is something that's used a lot in pancreatic tumors, especially gemcitabine and Abraxane in combination. And we use that here, a derivative of that is cisplatin. And we saw that there was something very interesting when we had the tumor in the beginning, very, very stiff in red. After 3 weeks of cisplatin with weekly cycles, we were able to see that part of the tumor, not the entire tumor, was actually softening. But there was a stubborn part of the tumor that was still pretty stiff. After 4 weeks, that stiffer area was receding, likely. But then when we stopped the treatment, that was the day 28, we had a resurgence of the tumor back to where it started. So that gave us a lot of hope of how we can better manage tumors and image them based on the underlying elasticity and separate responders versus non-responders. We're continuing that also in post-surgical specimens. We published that also last year, and it does translate to humans. So here's where you see that a tumor that has been not treated with chemo is actually much stiffer than the tumor that has been treated. And of course, the normal is the softest region. We didn't find a very big difference between with chemo and without in humans. So that explains why pancreatic tumors cannot be properly treated. So maybe that could help evaluate why some of the drugs that are available in animals and work well do not necessarily translate to humans. Okay, the last part I want to talk about is focused ultrasound, which is basically the therapeutic ultrasound, where you have this curved aperture and you're able to perform the effects that I showed you with ablation locally in a very focused manner, hence the name, so that you can change with the frequency or the wavelength or the actual geometry of the transducer. So you leave everything else intact, and in this very small millimeter-sized region, you can mechanically change or ablate the tissue. So we're doing that, of course, in the same mouse model. Once we identify the pancreatic tumor, we're able to actually ablate, and we can see how even during ablation, the displacement goes further down. As I said earlier in the breast cancer discussion, that causes further stiffening, and that informs the fact that we have successfully ablated a tumor. But I want to talk mainly about the brain in this very last part of the talk. We do also mechanically open the blood-brain barrier, so no ablation here, for the treatment of Parkinson's and Alzheimer's. And I'm briefly going to end with some of the neuromodulation aspects. So in the case of blood-brain barrier opening, we inject microbubbles, which are used as contrast agents in ultrasound imaging. And then we apply ultrasound. And of course, the microbubbles are micron size. The ultrasound is megahertz frequency. So they're resonators. The microbubbles become resonators, engaging the surrounding endothelial cells and allowing the loosening of the blood-brain barrier and the diffusion of the drug. Now, at the same time, they allow diffusion of contrast agents. And in MRI, we can see that the gadolinium, which is a contrast agent that's used in the brain, normally does not permeate the blood-brain barrier unless it has been opened. And we can see two regions that we opened here in the mouse, 3D rendering, and of course, MRI for safety shows no edema or hemorrhage in those regions. And when we go to the really, really high magnification level, so 20 microns is the scale bar here in confocal macroscopy, we see actually that the tight junctions, which are the proteins that are linking the endothelial cells, are actually not necessarily disrupted. So we can engage some transport through membranes of the cells here. So the tight junctions are in green, and you can see the permeation here of a fluorescent agent, biocytin. And most importantly, we can reach the neurons. This is a transduction of a gene delivery in a neuron where it expresses the green fluorescent protein, and we're able to see that this is successfully expressed in the neuron here. And co-localizes very 95% with neurons, leaving the surrounding cells intact. So once we know that we can get some pharmacologically relevant compounds in, now can we treat neurons? And we use the model for Parkinson's here called MPTP, and we injected first the toxic agent to deplete the neuron, and then we open the barrier and use a neurotrophic factor to replenish the neuron. And then we waited for 21 days to allow degeneration, open the blood-brain barrier, and again, as I said, deliver the nutrient once or 3 times. And then we saw how much of the neuron survived the insult. So I'm going to go a little bit fast here. It's a lot of histology. Just to show you that the neuron here in brown is much better survived in the region where we opened and delivered the protein, as opposed to the contralateral side, both with a single weekly session or with a 3 weekly sessions. And we're able to increase the cell-body ratio in the region of the dopaminergic neuron with the 3 weekly sessions, but also the dendritic fiber density increased both with a single session and the 3 weekly sessions. So on the other side of the neuron are the terminals. We were able to replenish those only in the 3 weekly sessions. 1 session was not enough compared to the contralateral side. So overall neurorestoration upwards to 50% happened here. So with 3 weekly sessions, we were able to get restored part of the neuron, the dopaminergic neuron in a Parkinson's mouse model. Now, can we do that also with the gene delivery as opposed to protein delivery? The answer is yes. I showed you earlier that we were able to reach the neurons with green fluorescent protein. Here's where we actually can reach them with the actual neurotrophic factor, and you can see the dendrites here in the pars compacta region of substantia nigra are able to seen as a higher density, depleted, I'm sorry, on the other side when we only have the drug only. And for Alzheimer's, very similar effect, but without any drug, just the opening alone elicits an immune response that reduces the tau protein. That's one of the proteins that is highlights Alzheimer's, both in the contralateral and ipsilateral hemisphere, as it turns out. And we have linked that to the immune response, as I said. So these are the microglia in the brain that are typically there to reduce any toxic proteins. They're not necessarily alerted to the fact that there is tau protein unless ultrasound is applied. And we see here that they get activated by 4% higher than without ultrasound, and they lead to reduction of these toxic proteins. We have a nice correlation here showing that. And the way that they reduce the actual tau protein is through phagocytosis. That's absent in the case of the without ultrasound, and again migrating to the site where the tau is, they're able to phagocytose and reduce it. And of course, that also is linked to working memory improvement. So after you reduce the actual amyloid plaque that you see here in another model, in this case it's amyloid instead of tau, you're able to see that the mouse is able to find the platform in a Morris Water maze or working memory test than the mouse that's compromised memory-wise. Okay, can we move this to the clinic? And the answer is yes. We've actually done our first Alzheimer's patient as of last month. We use targeting with MRI. We plan with CT and wave propagation models. We're able to monitor the bubble activity called cavitation and then assess after the fact with the MRI. This is what a clinical blood-brain barrier opening system looks like. So the patient sits here, we target with neuronavigation, and we're able to open the barrier within a few minutes and without any anesthesia. And then the patient goes to the MRI to make sure that we've opened successfully and also for safety. I don't think I have time for the modulation. I want to show you just the fact that with ultrasound alone, so no macro bubbles, we're able to map some of the motor areas in the brain versus sensory areas. The same thing we can do also in nerves, where with the EEG, we're able to elicit the suppression of pain and also image where the nerve is moving during focused ultrasound. I want to thank my group, our sponsors, and our collaborators, and you for your attention. Thank you.
No, thank you. Again, absolutely fantastic talk, very educational, and a lot of things touched upon. I don't know how you have the time to do all that work, to be honest.
It's because of these people here. I don't do all that work.
You have a huge team, which is awesome. Do you have time for questions? I know that you want to
I do have. I can take a couple, yeah.
Okay, let me just get to that. So we've got a question from Faisal, who's based in Dubai. He said, this was absolutely inspiring. Hold on. Absolutely inspiring, especially for students interested in research applications across medicine and engineering. What advice do you have for young researchers in joining such initiatives early on?
The earlier the better. If you are enthusiastic about work like this, then get into a lab and start working. And I know in the beginning, it's very hard. People not necessarily want to hire a person that doesn't have any experience or very little experience. Mm-hmm. But there's always something that people can do. We have high school students, we have obviously undergraduate students, but we're big believers that that you start early, and then a lab is an acquired taste. I mean, nobody's going to give you a map of a lab. Nobody's going to give you homework to do later. You will have to you have to find your way through. Yeah,
absolutely.
And go to a lab that nurtures that. There's some labs I think that don't have the infrastructure because there are few people. There are only a couple of PhD students, and they should be doing other things and training. But we have the luxury of having a big lab and a lot of more senior postdocs that are interested in teaching and doing and training.
Fantastic. So I hope that answers Faisal's question. I do have one question. So I liked, on your talk, you focused a lot on ultrasound. So do you think that obviously the future in terms of healthcare is to overcome the pathologist? Because at the moment, if we have suspicion of inflammation or a cancer, we take a tissue sample. But my understanding is, the work that you're doing, you're trying to use imaging for real-time diagnosis and then move on quickly to real-time treatment. Because on one of your slides, you mentioned that after surgery is done for cancer resection, you can then image it there and then to see if it's completely gone. So you wouldn't need to take biopsies, or is that the plan? Or?
That's the dream.
Of course.
Yes, we want to. Of course, we have a lot of work to do, but I think this is how elastography was born. In fact, almost 30 years ago now, is to replace breast biopsies, and I think some of them are spared. Not all of them, of course, because there are some tumors that are very inconclusive. But I'm really excited about our work in chemotherapy, because in chemotherapy, during chemotherapy regimen, you cannot necessarily biopsy the tumor. You can biopsy the diagnosis and maybe at the end of the chemo, but when you do, monthly sessions, and it's much harder to do. So, and I think especially tumors such as triple negatives or pancreatic tumors that I showed you, nobody understands why those tumors come back, or they're not treated properly. So I think imaging has a lot to offer, from not just taking a sample, but as we showed in tumors, where it doesn't work. So then you keep the drug for where it works, and then you add another drug to basically be able to have a tumor suppression where you need it. So you can sample that area, understand the genetic signatures, for example, of this tumor, and then attack this region, because it could be 2 different mutations, for example, that you could be dealing with. So I think using radiomics, as they call them, right, so to just try to inform genetics and vice versa through imaging, I think it's the future for doing this better.
Awesome. Time for one more, or?
Sure.
Okay, so we have anonymous, not revealing, with microglia going through assistance then destructive phase. How do you monitor this retreatment?
So, okay, that's a very good question. So microglia, absolutely, they can be a double-edged sword. They can phagocytose, as I mentioned, and then the actual toxic protein, but they can also take, in the case of advanced, for example, Alzheimer's, they are attacking the blood vessels and attacking the neurons. So the only way to curb that is to understand it. And there are what's called pro-inflammatory cytokines and anti-inflammatory cytokines, the chemokines. And we're looking at that very, very closely. Is something that my lab, I'm an engineer, so, all of a sudden I have to understand biology, which is basically what we're talking about, right? You can't really completely say, I don't want to understand medicine, I don't want to understand biology, I just want to, put out a good image.
Yeah.
So the one thing is that we have experts. Otherwise, we wouldn't be able to do that. We surround. Columbia has a huge neuroscience, neurology group, very, very active, and immunologists who guide us through it. And with qPCR understanding of what happens to this cytokines and chemokines, together with RNA sequencing, single cell, and all that good stuff, you're able to sort the cells one by one and understand which one gets activated, how, where do you get peripheral cells to come in, where do you get resident cells to come in. Does it get complicated? Absolutely. But this is the only way to say, this is what we don't want to cause any more problems than Alzheimer's or Parkinson's or other diseases are already causing. We just want to treat it. And there is a way right now where this renaissance time, I think, of biology, we can take advantage of that and say, not all cells are the same and not all cells activated are the same. Let's sort it through.
Perfect. Great. Okay, well, thanks again for your time. I know how busy you are, so we really appreciate it.
Thank you for having me. I'll check it out later after I get to the lab.
Yeah, awesome. Great. Thanks very much, Elisa.
Thank you. Thank you. Bye, Neil.
Bye. Okay, so we're going to take another break. Our next speaker will be coming at 3 p.m. UK GMT. So again, stretch your legs. You don't want to develop any clots while sitting for this length of time, and have some fluids, also fruits. We'll see you at 3 p.m. Rebecca, can you hear me?
I can, yeah. 1 second, I'm just trying to sort out my sound. Can you hear me?
Yes, we can hear you, yeah. Take your time, don't worry. We're doing a live event, so you are allowed. Okay, great. Okay, hi everyone. So we're back for our third speaker of our Clinician Engineer Hub conference. As you know, we've brought in the best speakers across academia and industry over 3 days, completely free of charge, and everything will be recorded. For those of you that are busy at work, you can view it later. So now we have Professor Rebecca Shipley. I'm sure everyone that knows engineering and healthcare in engineering knows of her. She heads up University College London. She's a mathematician in terms of background, and she focuses on lots of elements of engineering, which I'm sure we'll hear about later today, but around tissue engineering, vascular elements, and also nerve engineering. And I think that you recently headed up the Nerve Engineering Center at UCL, among other accolades. So I will let you begin your talk, and then we can take questions and answers afterwards. So Rebecca, if you can share your screen.
Super, thank you very much.
No problem.
How's that?
Perfect.
Can you see that?
Yeah, fantastic.
Brilliant. Well, thank you ever so much for the invitation to come and talk to you all today, and for the kind introduction. As Neil said, I'm Rebecca Shipley. I'm a Professor of Healthcare Engineering at University College London and also Director of our UCL Institute of Healthcare Engineering. And today I'm going to start by telling you a bit about the healthcare engineering community at UCL, and then talk through a specific case study of what we've been up to this year, which is around the manufacture and deployment of non-invasive breathing aids for COVID patients. So particularly topical and relevant for everything that we've been facing this year with the COVID pandemic. I thought I'd just start by telling you a bit about our Institute of Healthcare Engineering, which I direct. We're essentially a community at UCL which interfaces our engineers with our medical science and clinical science communities. So we're, I think, particularly fortunate at UCL that we have a very multidisciplinary collaborative culture. And this institute is really dedicated to developing digital and medical technologies. We have a really vibrant engineering community spanning all the kind of classic engineering themes that you might expect, like mechanical engineering, electrical, chemical, biochemical, computer science, etc. And then UCL's also very well known for our medical sciences community and our connections with our partner hospitals. So we have really strong links with a whole network of different hospitals and trusts across London. And we really aim to collaborate with our clinical communities so that we have a really strong understanding of the unmet clinical needs and can then develop the technologies to meet those needs. So the Institute has 5 main themes which really span the care pathway, from prevention, monitoring, diagnostics, therapies and interventions to rehabilitative and assistive technologies. We have a suite of research priority areas, which are really the themes that we are seeking to develop over the medium term and which we think are really important for long-term health and healthcare. So I've listed those there on the left-hand side. They're self, shared, primary and community care, timely detection, diagnostics and intervention, healthy aging and multimorbidity, and effective and cost-effective technologies. And today I'm going to be telling you about a specific kind of case study, if you like, of how we've tried to build and deliver on this community in response to the COVID crisis. Another initiative that I just wanted to quickly tell you about is our recently launched Age Innovation Hub, which is essentially a kind of crowdsourcing and engagement platform to engage with the public and with healthcare professionals to understand people's experience of aging and the kind of technologies that they would like to have in place to support healthy aging. So with this hub, we're currently going through an ideas generation phase where we're seeking input from these kind of communities, and then into next year we'll be taking forward and funding specific projects and technology development plans that come out of it. I think that hopefully gives you a bit of a flavour for what we're about in this space at UCL. If you'd like to find out more, there's a lot of information on our website, and you can follow us on Twitter as well, so please do get involved. Right, so today I'm going to focus on telling you about what we did at UCL this year in response to the COVID pandemic, and specifically around ventilation devices for the NHS and internationally. Now, a lot of you might have heard about the need for ventilators and ventilation devices to treat COVID patients, but we're going to focus on a non-invasive breathing aid called a CPAP. So I'm first going to introduce to you what a CPAP is, and then we'll talk about how we designed, manufactured and delivered them for the NHS and more recently internationally. So a CPAP device, what is it? A CPAP stands for continuous positive airways pressure, and it's essentially a device that takes pressurized oxygen, for example, from an oxygen port in a hospital, and delivers it to a patient through a face mask. And the idea is that it essentially provides a positive pressure which splints open the lungs and the lung bases and enables you to get more oxygen into the lungs than you would do using just a face mask or through normal breathing. So essentially the role of CPAP is to support a patient's breathing. You can provide an air oxygen mix with where you can control the amount of oxygen or the concentration of oxygen in the inhaled air. And as I said, that's delivered through a positive pressure, either through a mask, as in the schematic on the left-hand side here, or through a helmet, which is basically a hood that sits right over the patient's head. These aren't new. They were first invented over 100 years ago, and they're used in commonplace in respiratory therapies or to treat acute respiratory failure, through to also sleep apnea. So they're not new pieces of kit. They're devices that are used commonplace in respiratory health in the UK and internationally. So what was the need for CPAPs for COVID patients? Well, I think you will probably all have heard about the need to provide oxygen and respiratory support to patients who admit in hospital with COVID. It's the most common reason that people need hospital treatment, and essentially the problem is that they're not getting enough oxygen into their bloodstreams. Now, there's a lot of discussion about the use of ventilation or mechanical ventilation to treat COVID patients. And indeed, looking back to kind of mid-March, you might remember in the UK that the government launched their ventilator challenge to kind of corral the UK engineering and manufacturing community to design and mass manufacture more ventilators to treat COVID patients. Now, looking back at that time, I have a strong collaboration with a chap called Mervyn Singer, who's a professor of critical care at UCL and is also one of the lead consultants at University College Hospital. And he was talking to intensivist colleagues internationally, so particularly in China and Italy, to understand what their experience had been of treating COVID patients. And at that time, the essential care pathway for treating COVID patients was if they needed more oxygen than you could deliver using a face mask, then to mechanically vent a patient. So mechanical ventilation involves sedating a patient. They're usually sedated for potentially weeks at a time, and the ventilator essentially breathes for the patient. So it's highly invasive and is obviously really critical for the most sick of COVID patients. However, the experience in Italy and in China and then Italy was that when they moved straight to ventilating patients, they quickly found that their healthcare systems became overrun. And so they started adopting a CPAP strategy where they use CPAP as a non-invasive breathing aid as an alternative bridge to ventilation, and it was keeping around 50% of patients from progressing to needing ventilation. And that was a really positive impact, because not only did it enable optimization of the available healthcare resource, so the availability of ventilators and trained staff to use them for the patients who needed it the most, but also it was good for patient outcomes, because essentially if you don't need to mechanically ventilate a patient, if you can avoid that, then obviously that's preferable. At that time, back in March, UK hospitals had a very limited supply of ventilators, but also CPAP devices that could deliver the required oxygen concentrations. So there was an unmet need in the UK to develop and deliver a simple CPAP device that could be approved by the regulators and mass manufactured at real speed. So this was looking back to mid-March, where the first wave was clearly developing at pace, especially in London within the UK, and then spreading across the rest of the country. And so we needed to be able to deliver these devices within weeks. So we formed a consortium at UCL that spanned UCL engineers, critical care experts, both at UCL and at our sister hospital, University College London Hospital. And then we also partnered with various industry players. So the lead industry player was Mercedes HPP, high performance powertrains, and they're the the arm of Mercedes who design and manufacture the Formula One engines for Lewis Hamilton, for example. So they're real experts in gas flow systems, in precision engineering and manufacture, and they're also pretty good at doing things very quickly. So we formed this consortium, and our objective was, as I said, to be able to design a device that could be manufactured at speed and rolled out across the healthcare system. And that's essentially what we did, and I'm going to try and tell you a bit about that story today. So just showing you some of these pictures, I don't know if you can see my mouse moving around, but here you can see some of the team. So the chap in scrubs, as you might expect, is the clinician. So that's a chap called Dave Brearley. Dave and Mervyn Singer were really the critical care or clinical leads. Tim Baker here was my co-engineering lead. He's a colleague at University College London, and previously to being a prof at UCL, used to work in the motorsport industry, so had this really excellent network of links in the automotive industry. This chap here is Andy Cowell, who's the MD of Mercedes HPP, and this is Ben Hodgkinson. So they were essentially the leads from Mercedes. Now, this is the device that we ended up manufacturing. As I said, our focus had to be on a simple device so that it could be made very quickly, and B, would be easy and straightforward to train healthcare workers to use. And it also needed to be something that could pass the regulator approvals quickly. So what we did was we started from a historic device, the Philips Respironics Whisperflow, which was an off-patent device that had been used widely in the NHS and internationally up to about 10 years ago. The benefit of it was that it's very simple. It's a purely mechanical device. There's no embedded electronics. And also there was a wide clinical utility and safety history that was obviously beneficial to accelerating through regulator approvals. So we formed this consortium. We essentially went and found one of the old Whisperflow devices in the Anaesthesia Museum at University College Hospital. And sadly for the clinicians, the first thing we did was cut it up, basically understand exactly how it worked. It's essentially works on the Venturi principle. So if you look at this drawing, well, this picture here, sorry, this port on the top right hand side plugs into the pressurized hospital oxygen supply in an individual ward. Here's an on-off switch. This button here, or this knob here, lets you set the flow, so you can tune the flow rate, and this lets you tune the oxygen concentration in the inspired air. This is the air entrainment port, and then this connects out to the patient via a series of tubes, filters, and masks. So essentially from our first meeting, we were able to basically take this apart, do all the analysis on it, redo the 2D, 3D drawings and material analysis, and then fabricate at Mercedes. They made the first prototype within 10 days. So from our first meeting, sorry, within 100 hours, from the first meeting, we were back in the hospital testing this on healthy volunteers. We then subsequently submitted it to the regulator, the MHRA, who are the medical regulators in the UK, and received approvals for its use in the UK within 10 days. Now, at the time, there was real concern about the demand for oxygen therapy in hospitals. So there was real unprecedented need to supply oxygen to patients, and given the volume of patients that were being admitted to hospitals, there were real concerns whether hospital oxygen infrastructure might cope. So mindful of that, we did a lot of work on a version 2 of this device, where we essentially sought to minimize its oxygen utilization. So we did work, for example, on the design of the air entrainment port, and also coupled to that, the design of the breathing circuits, which connect this device to the patient and offer resistance to airflow, essentially. That combined design process enabled us to reduce the oxygen utilization by up to 70%. We then submitted that Mark II device to the regulator and received approvals for that. And then working with Mercedes HPP, went into mass manufacture. Alongside this, one of the really critical things to think about when you're developing any kind of piece of medical technology is how it will be used in a hospital setting. So alongside this, we developed a platform of training materials led by our clinicians, including user and clinical guidance documents and training videos, and released all of those alongside the device. Excuse me. This is just to give you a bit of a feel because I'm conscious that I'm telling you a story and maybe not so much about the engineering. So I thought I'd better tell you a bit about the engineering detail. But this is some of the work that we did to essentially do the reverse engineering piece and the design optimization piece. So on the left, you've got examples of CT images done by the Mercedes teams of the Philips Respironics whisper flow sections, so that we could do full material characterizations of the device. And on the right here, you have some of the 2D drawings that we used in the manufacture. On the right-hand side, you've got some examples of the computational fluid dynamics work that was done to essentially refine the design from the Mark I to the Mark II. So this was looking specifically at modifications to the inlet geometry, an air pump, which enabled the reduction in oxygen utilization. And then we also did extensive bench testing. So, pivotal to our MHRA submissions were in the first instance demonstrating exact like for like with the original Philips Respironics device and for the Mark I, and then subsequent design iterations. So this is all bench testing work that was done to essentially show parity in how our device operated compared to the original Philips Respironics one. We obviously then went forward to full healthy volunteer evaluations at the hospitals and then clinical trials. Now, I mentioned the mass manufacture piece. So once we had approvals from the regulator, we didn't have long. So the target was to be able to deliver these devices to the NHS in time for the peak of the first surge, which was due to hit in London by Easter weekend. So we started in mid-March, and so we had weeks. Now, this is where the Mercedes expertise really came into play. So we ultimately got an order for these devices for 10,000 of them to be delivered. Well, the order was from the Department of Health and Social Care, and obviously to be delivered into the National Health Service. Mercedes at the time were stood down, so the Grand Prix, Formula One Grand Prix was paused because of COVID, so they had capacity, and they repurposed their whole plant basically at Brixworth and dedicated it to this manufacture. At the maximum rate, they were manufacturing 1,200 per day, which was really phenomenal. We then partnered with GTEM, who these photos are of. So they're essentially an automotive just-in-time logistics company. And we partnered with them to do delivery to the NHS. So they would essentially pack and deliver kits that comprise the CPAP device, counterpart oxygen analyzers, which I don't have time to tell you about today, but we had to go through the same process of design and manufacture for oxygen analyzers and breathing circuits. And essentially they would deliver them the next day to any hospital in the NHS. So again, they were just phenomenal partners. And this gives you a feel for the kind of team that we had behind us. So pretty much everyone in that bottom photo was dedicated to manufacturing of these CPAP devices. So they were really supporting and doing a lot of the engineering work with us, but then also doing the mass manufacture. So we were really privileged to be able to work with these teams. This schematic really summarizes the timescales that we were working to. So I mentioned that our first discussions started in the middle of March. So we first met, so myself, Tim Baker and Mervyn Singer, the lead clinician. We went for a drink on the 17th of March. This was as lockdown was coming in, and really defined the focus on CPAPs rather than mechanical ventilators, and the initial starting point of reverse engineering the Philips Respironics whisper flow. So the Mercedes teams joined us at UCL the next day. So we rang the Mercedes teams that evening to ask if they would be interested in collaborating. We already had an extremely strong link with Mercedes HPP through UCL Mechanical Engineering, and a team of 3 or 4 of them arrived the next day. So from that first meeting, so that was a Wednesday, on the Sunday, we had the first prototypes. So they were manufactured at Brixworth, and the Mercedes teams drove them down, and we were back in UCLH that evening, testing them in the hospitals. That was a really memorable process, because at the time, the hospital was essentially empty. It had been prepped for the COVID patients that were, already arriving, and the anticipated numbers. We were essentially testing these devices in different wards and settings to see if they worked as we would expect them to. So after that kind of platform of healthy volunteer testing, We submitted the designs to the MHRA to receive regulatory approvals alongside a plan for clinical evaluations, which we did on COVID patients at UCLH and 3 of our sister hospitals. And then that went into mass manufacture. So by the 15th of April, we delivered the 10,000 devices and they've been distributed across the NHS. We also released all of our designs and manufacturing instructions at zero cost on the 6th of April to support the global response. And I'll tell you a bit more about that in a moment. So what has the clinical impact been? Well, so far, the CPAPs have gone out to over 160 hospitals across the UK. So that's through the National Health Service. So it includes England, Ireland, Scotland, Wales, and also some of the overseas territories like Montserrat. And now the clinical user data has been in alignment with the initial findings from Italy and China that the use of CPAP can reduce the need for mechanical ventilation by about 50%. In general, the kind of broad clinical experience is that patients can be managed safely on CPAPs outside of an intensive care setting, so they can be managed in respiratory wards, and that, you know, if you can treat a patient using CPAP rather than mechanical ventilation, the outcomes are good, patient turnaround time is faster, so the length of stay is shorter, and the complication rates are much lower than using mechanical ventilators. So for example, there's a high incidence of renal failure and pulmonary issues for patients on mechanical ventilators, which you don't see on CPAPs. So if you can avoid the need for a patient needing to progress to needing mechanical ventilation, that is good for patient outcomes. There were also initial concerns around the risk of infection spread. So obviously CPAP involves pressurized oxygen, which can then aerosolize and spread droplets. Those initial risks haven't been borne out based on environmental testing by Public Health England, but there's a real critical need for sufficient and high quality PPE to be in place for all healthcare workers. In terms of specifics of clinical impact in the UK, now we never did a big clinical trial, but if you look back for the ICNARC, which is the Intensive Care National Audit data, for the UK over wave 1, the mortality rate of patients in UK intensive care units fell by about 21% over that time, and that coincided with a reduction of up to 26% in the use of mechanical ventilators. Now, obviously, there's not cause and effect there. There's many other contributing factors that link to that mortality rate, not least the use of steroids, for example. But broadly speaking, there's been significant uptake in the use of non-invasive ventilation to treat COVID patients, and positive outcomes for both patients and for managing hospital resource. So finally, with my last bit of time, I'm going to tell you about some of our work to try and make this technology available for the global response for COVID. Now, back in March, April, when we got these regulatory approvals, we received quite a lot of media interest for this work, and we started getting a lot of approaches from other teams in other countries to understand how they could get access to the technology. We decided as a consortium that we should release all of the designs and manufacturing instructions at zero cost. We never protected IP for this. We always saw it as a humanitarian project, so we weren't seeking to have any financial gain from it. Far from it. We saw it as a contribution to you know, the global response, essentially. So on the 6th of April, we made all of our designs and all of the manufacturing data freely available through a zero cost license. So the license is free, but it essentially is it's a legal license which transfers the legal liabilities and regulatory considerations to the holder of the license. They're available to manufacturers, to research institutions, healthcare providers, and the non-profit sector. And the release really gives us every piece of information that we could put together. So it's the full manufacturing drawings, all of the system characteristics and schematics, the full bill of materials and tooling information, all of the test protocols, assembly procedures, and data that you should be able to match. It's been very popular. So we've approved around 1,900 downloads of the license, which spans 105 countries. which has been really exciting. And there's a whole range of different teams in different countries that have made different levels of progress with manufacture in country. And I'm just going to talk through a few examples, but to give you a kind of spectrum, there's around 20 teams now who are progressing and are able to manufacture these devices in significant volumes in country, and are getting regulatory approvals, doing their own clinical trials and clinical rollout. And that spans a whole suite of countries in Latin America. Other examples are Australia, Canada, Germany, and then India, Pakistan, South Africa, Russia, and others along the way. Now, we didn't have any experience of doing this when we set out, so we were kind of learning as we were going, and I think we've learned a lot along the way. I think one thing that's really important whenever you're, as an engineer, developing a technology is that it needs to be done in really close partnership with the end user of the technology. So you need to understand how the technology will work in a clinical setting, and a whole suite of more the human factors, if you like, so that determine how people engage with the technology, what the kind of setting is. Is there access to power? Is there access to breathing circuits? What's the regulatory environment? What's the training environment, etc., etc. Now, learning as we kind of went, we've put in place as much as we've felt able to provide support around a whole suite of different factors. So we have a technical and manufacturing support team who essentially answer questions around anything to do with design and manufacture. We've got a Facebook group that connects manufacturers in different countries. The IMechE have been just phenomenal. So they were phenomenal in being so open and collaborative when we were doing the work in the UK, but once we've moved to this international work, they've also provided regulatory support to regulators in different countries to understand the IMechE processes and how that can be supported, adapted in other countries. We've done a whole series of clinical webinars, and there's a clinical essentially support team. So we've done webinars to clinicians in different countries to support training locally, and we've published all of these webinars online alongside all of the training materials, the user documents, etc. And we've had those translated into different languages where we've had the resource and expertise to do that. And the other component that we've done a lot of work around is local supply chains. So looking back to March, when COVID was really spreading at pace across the world, I think around 80 countries imposed export bans on medical products. And that really inhibited countries who rely on import to get access to medical kit. So one of the restrictions and challenges for many countries has been just access to things like consumables, to medical devices, to drugs, etc. And then, alongside from that, access to the kind of materials that you might manufacture these devices with. So we've worked with different teams, particularly through the UK Department of International Trade, to provide as much support as being possible to local manufacturers, and that, that notwithstanding has been a challenge. I should actually also mention that one of the real considerations for these devices is the availability of therapeutic oxygen. In the UK, all of our hospitals have got a piped oxygen supply, which provides pressurized oxygen to individual wards. When you look at that globally, that's not far from universally true. So if you think of many developing countries, in particular in more rural settings, there isn't necessarily access to piped oxygen. There may be access to cylinders, oxygen cylinders, which these devices can run off, but obviously the oxygen flow rates can be prohibitive. And then, there's many settings where there isn't even access to oxygen cylinders. So again, understanding whether a device is appropriate for a particular setting is really needs to be discussed and considered alongside the clinical teams who are working in those settings. And but we're by no means claiming this is some panacea that will solve solve COVID in all places. It's far from that, and it's, you know, where it has provided, I think, real benefit is where there is oxygen infrastructure in place in kind of established hospital settings in low middle income countries. So I'm going to provide just a few examples of different countries that have manufactured themselves. So the first one that came in was Canada. I think they produced their first prototype by May. Mexico were one of the Latin American countries that came next, and you know, there's a whole suite basically of automotive companies in Mexico that came together to do mass manufacture, and they've now progressed excellently. They're making hundreds and have used them in hospitals across the Bay of California region. These are devices that have been manufactured in Peru. So again, Peru have made really fantastic progress, both in terms of manufacturing in country, and then have done clinical trials across various different hospitals in Lima. This is a prototype that has been made in Kenya, which again was actually one of the earlier countries to succeed at prototype manufacture. And here's a few more. So on the top left here is an example from a consortium in India who have now not only completed manufacture, they've got regulatory approvals in India, and this is an example of one of their manufactured devices being used on a patient in India. I mentioned Peru already. A lot of the Peruvian work has been done in very close collaboration with the Peruvian embassy in London, who have really supported both the clinical and manufacturing teams in Peru, and as an interface with us to provide the kind of support packages that we've got in place, and provide things like access to some of the materials and breathing circuits which are needed. And then on the top right, I think that's a video, but I'm not sure it'll work. Or maybe it will. Oh, sorry. That's in Pakistan. So again, there's a consortium that have gone all the way from manufacture through to regulatory approvals and clinical rollout in Pakistan. And as an example, they just donated 500 to the Tajikistan army. So the final thing that I'll quickly mention for a couple of minutes is our work, in particular working with the International Medical Education Trust to deliver devices directly to countries who essentially don't have the manufacturing capability to be able to manufacture at the pace to meet COVID. So where the manufacturing piece wasn't an option, we've sought to try and work with individual charities and governments to provide the devices, again working on a non-profit basis. So a particular example I'd like to refer to is, as I said, this work with IMechE 2000. This is a charity who works in medical education and training and device distribution across Palestine, Africa and Asia. And we've worked with them on two main examples. So first of all, in Uganda, so we've supplied devices to Hariri Hospital. This is a patient on one of our devices in Hariri Hospital and Lakore Hospital. And then also to Palestine, where we've partnered alongside with IMechE with medical aid for Palestinians. And this has been an example where I think it's really been able to flourish and succeed because of the strong partnership with clinicians on the ground. So we helped through MAP and through IMechE to contribute to clinical training programs. The devices, this should now be 30 actually, but 30 devices have gone to across 5 Palestinian hospitals. But MAP and IMechE have done a clinical training program reaching 750 doctors and nurses. And that's really enabled the clinical uptake. And you know, we continue to raise funds to try and deliver more devices to these hospitals and to more hospitals in Palestine where there's a really urgent need. I'm going to stop there. Thank you so much for your time. I hope that's been interesting. It's a kind of case study of what we've been doing this year. It's been a real privilege to work on this project. There's a whole host of acknowledgements. I couldn't possibly thank everyone, but not least all the people at UCL and UCLH. Here are industry partners. So I mentioned Mercedes HPP, but GTEM, Intersurgical, Life Racing, and the government teams that we've worked with as well. We've got some papers coming out. So this one in Lancet Respiratory Medicine tells the story if you're interested in reading more. Thank you very much.
Awesome. Thank you very much, Professor Rebecca. That's great. You are probably, I would say, the first speaker that I think you've blown away the audience, because there's not a question. And I think the reason for that is that the way I look at this, from the start that you wanted to do this project to the end, everything just flowed perfectly. Did that surprise you, or? I mean, I just think it's incredible, because you have the concept, you managed to partner with industry, clinicians, everything from the get-go in, I think you said in April, and then what you've rolled out is just, well, I would say insane in a very good way.
Oh, thank you. You know, it would be unfair for me to say that it's been that straightforward. So, you know, I'm you know, obviously very happy and proud of what we managed, how far we managed to get, but, you know, it certainly wasn't easy at times. And, you know, I think there's a whole host of components that were really, you know, challenging and very stressful at times. And obviously there were real time pressures.
Sure.
And, you know, we wanted to be able to do something within a timescale that would be valuable to the NHS first and then internationally. So I think one of the real things that's worked well for us is that we've had this multidisciplinary team. So, you know, our team was clinicians, engineers, manufacturers, but we were, you know, the sub team that were kind of the decision makers. We were, you know, there's about 6 of us, and you know, we met daily. Well, we were communicating continuously. But it was a genuine partnership of equals. There was no one person dominating over anyone else, and everyone respected each other and supported each other. And you know, it sounds probably a bit cliche, but I think for these kind of projects where
it's essential,
there's technology development and there's clinical impact, that's the only way that it can work, really.
No, 100%. I have another question as well with the regulation side of things. Obviously, you don't need to go into all the details, but in a situation like COVID-19 and the pandemic, my guess is that there is a fast track regulation, or because I think when I looked to your schematic, when you developed the device or the prototype, you showed that it was successful. It seems that it then rolled out within 2 weeks, something like this, or?
Yeah, absolutely. So the MHRA, so I mean, so first of all, in kind of normal times, to develop a medical device and get through to regulatory approvals would normally take, I mean, years even.
Yes.
The MHRA basically repurposed, they redefined their regulatory process for the emergency of COVID. So essentially you can apply for special authorization for a device for use on COVID patients. So it essentially applies to non-CE marked devices and restricts the use to COVID patients. But it was really amazing, because essentially everything that would normally be in an MHRA submission was still in there. So we still needed to do, you know, all the bench testing, healthy volunteer studies, clinical evaluations, adverse event reporting, all of the pieces, you know, quality management processes, all of that was still in the dossier that we needed to deliver. But the MHRA were just phenomenal. I can't underestimate really how brilliant they were. So we approached them on day 2. I first reached out to them. And I think from that day onwards, I literally spoke to them at least once a day, if not like multiple times a day. So they were really supportive, collaborative. I think they were, the need was clear, the like clinical need was clear.
Sure.
So they were very happy to engage with us and provide real clarity on what we needed to do, if that makes sense. And that really accelerated that process. I mean, the other thing obviously was that we were starting by reverse engineering an existing device. So our Mark 1 submission was to replicate exactly a device that had been used for decades in the NHS and had, you know, very well evidenced safety and efficacy base. So, you know, for our Mark 1 submission, as long as we could demonstrate parity of performance, that provided that added security, if that makes sense. And then for our Mark 2, we were doing design modifications to that Mark 1 device to minimize oxygen utilization. So that kind of process and structure also facilitated the speed of getting through the regulators.
Very, very streamlined, exactly.
Yeah.
I have a question if you've got time from the audience. So this is from a medical student called Hamza. Thank you for such a fascinating talk. As a medical student in London, is there any way for students like me to get involved?
Yeah, always. So, so yeah, absolutely. I think, you know, one of the really exciting things that you can work in as a medical student or an engineering student is in these kind of medtech clinical engineering projects. So basically, if you'd like to get involved in anything, get in touch. I don't know where you are in London, but you won't be far from UCL. So yeah, we always welcome links, and contacts, and, you know, building of the community. So yes, please drop me an email.
Okay, fantastic. Well, thanks again. I'll let you head off because I'm sure you're very busy doing lots of exciting things. Our next presentation will be at 4 p.m. and that's Prof. Mughal, also from UCL and the Cleveland Clinic in London. So we're just going to take a break now. And again, I always say stretch your legs, have something to eat and drink, because obviously sitting down for this length of time is not easy. Okay, we'll be back at 4 o'clock.
Thank you.
Thank you. Prof, can you hear me?
Right.
Oh, perfect. Yes. Okay.
How
Good
are
to
you?
see you after so long. Yes, yes. It's been a very interesting day. We started at 1 o'clock, so coming to a close.
Right.
So I'll just introduce you and then we're ready to go.
Yeah.
Okay. Hi, everyone. So this is back to our first day of the Clinician Engineer Hub virtual conference. The next speaker is Prof. Mughal. Now, actually, a backstory to this. Now, as a medical student many, many years ago, Prof. Mughal was my first supervisor back in Manchester when I was training there. So I thought I would return the favor and get him to deliver a great talk for all the training he's delivered to me. Now, a bit of a background, Prof. Mughal is a surgeon, a fantastic surgeon. I can definitely vouch for that, based at UCL, and is also now the Director of Surgical Oncology at the Cleveland Clinic in London. So this morning, we've heard from engineers in the US and also engineers just recently, Prof. Rebecca Shipley at UCL. So now we're going to get a clinical perspective, surgical perspective about bridging the gap between surgery, medicine, and engineering. So Prof. Mughal, when you're ready.
Thank you very much, Neil, for that introduction. Can I screen share?
Yeah, you should be able to.
So we'll just do that in a minute. Let me get my presentation sorted.
Sure.
Okay. So let's see if it works.
Yeah, perfect. We can see.
Okay, great. Fine. Thank you. So I don't obviously know, Neil, we discussed this through email exchange as to what the content was going to be of the other speakers. So if I overlap with something that others have spoken about, please let me know.
Don't worry.
But Neil has given you my background. And one of the things that I have been involved with recently with the COVID pandemic until July was also managing this sort of response to COVID of, the cancer alliances. So I was a CMO for Cancer Alliance for this part of London, North and Northeast London. And one of the things I'm going to talk about arose out of that. So I think it just goes to show you how sometimes, in fact, quite commonly in adversity, you get innovation coming through. I thought I'd pick examples that I have personal involvement and experience in. So there is no direct link between these three, but at least these are things that I have been working on in a small or big way. The first thing is item there is phase contrast X-ray imaging, which I will explain from the clinical point of view. I bet there are people on the call who know a lot about the actual technology, which I can't tell you about, but I can tell you about the application we're looking at. And I think you'll all be aware, obviously, there's a huge profusion of apps for remote monitoring of patients' conditions and patient support, and I have been working in that area as well. And the last item there is tissue engineering. So I'm just going to say a little bit about that because I've also had some involvement there. Now, I don't pretend to be an engineer, but this is a very basic diagram which I can understand. So this is basically a synchrotron beam generator that you can see on the left. And you've got a specimen, a tissue specimen, which is in the middle there, and you've got a detector. And basically, with that sort of setup, you can get images to a resolution of 1 micron from tissue that is in there. So this just goes to show you an application of this which has been used. And this, I think it's a group in Southampton, and you can see that you got your source and you got lung biopsies that have been taken there, and you got a detector over on the right. Now, normally what happens when we take some tissue from a patient, when we've done an operation, we've done a biopsy, we take that tissue like you see there, it is then fixed, it is then chopped up into slices, and then it's stained, and then a pathologist with experience and expertise will look at it. It's all a manual process. All these steps are manual, and then we'll get a report that this is a cancer, or it's not a cancer, or it's inflammation, etc. And here you can see the equivalent pictures. On the right is a typical histology slide. These are the same biopsies that you've seen in my illustration earlier, and on the left is the X-ray imaging. Now, of course, the X-ray image is just shades of gray. So, you know, whereas with conventional histology, you can put in stains, which will be taken up by certain type of cells which stain differently. We don't quite have that in the sort of micro CT application. But, you know, one of the things that people are working on is to use sort of nanospheres, which can pick up tumors, and then hopefully that they will be able to be used in this technology to play the equivalent part of histocytochemistry, where you stain tissue samples to pick up particular type of cells. But you can see it's amazing resolution. And the other thing, of course, is that, and here's the real beauty of it. So if you see on the left, of course, whereas when you slice the tissue, you've only got slices that you can look at each slice at a time. On the left, you've got a block. So you can actually, you've got a 3D image there, and digitally, you can cut that image any way you want. So that's another advantage. And in fact, one of the things that this technology fascinatingly has been used for is to do autopsies of fetuses. So the fetus will obviously be very small. One of the limitations of this technology is that it can't process a large mass of tissue. But you know, you can do an autopsy of a fetus, and it'll give you pictures of the resolution which you need to be able to look at congenital heart defects, etc., in that fetus, or any other defects, without actually doing an autopsy in a conventional way. So there are some amazing possibilities. We got a grant from EPSRC. This is a joint collaboration with the engineering department at UCL in a few years ago. And we've been working with this technology to look at the esophagus, which is my area of interest. And as it happened, I just spoke to one of the collaborators this afternoon, and he sent me some images, which I will show you. So this is the esophagus. On the left, you've got conventional histology, and on the right, you've got the image by the device that our group is working with, and it is really quite amazing resolution. This is esophageal cancer, and on the left there is conventional histology. This is a lymph node with some tumor in it, and you can see it there. And there are some interesting results coming through. Now, what does this mean? Well, I've told you some of the advantages, which is that you can do 3D reconstructions, you can chop the specimen up in any direction you want to. But our aspiration is that we would be able to take out the esophagus, take out the esophageal tumor. And if the further iteration of this machine can be built so that it's in the operating theater, we can put the specimen in there and see whether any of these resection margins are involved in real time, and we can change the operation or the treatment at the time instead of waiting for 7 days after completing the operation, before we know what to do with the patient. So that's an aspiration. We're not there yet, but that's the direction we're going in. Turning to remote monitoring. So I mentioned about the pandemic. One of the things that has really exercised us everywhere in the country is how do we deal with patients who have got urgent non-COVID conditions. And for us, in my last role, it is cancer patients who were waiting for surgery. In London, we managed, thank goodness, to make these surgical hubs, which were centered in the private independent sector, so that we did not have patients coming as emergency with COVID. So we had a secure environment. But of course, the point is that you wanted to decrease the footfall, with the patient not coming back and forth from the hospital for follow-up visits, etc. So I'm going to just tell you about some of the work that one of our urology sort of fellows had been doing, which was on remote monitoring of patients who had undergone a big operation to either remove the bladder or the prostate. These are now done robotically. Now, the great advantage of these operations is that patient only needs to be in hospital for a day or two. But of course, the downside is that they might still go home and develop a complication there. And is there a way that we can monitor these patients so we get the heads up about complications that might be in the making? And so what was done in this pilot study was to develop a Fitbit type of device, which was able to look at the patient's pulse rate, also measure oxygen saturation. You had a device that measured the temperature. You had a device, it also tracked the patient's steps, etc. And what you've got here is a specimen there. So you got surgery, you got discharge. So this is actually 7 days after the operation, rather bigger operation. And you got here basically the device registering a heart rate, which is nice and stable, step count, which is stable. And here on the bottom, you got a situation where you can see that things are all right up to about that point, but you now get a situation where the step rate goes down, the heart rate goes down, and in fact, the patient actually only pitches up at the A&E on day 12. But you can see that with this device, you could actually sense that there was something that was brewing up several days before this happened. So this is now undergoing sort of testing the proof of concept in a larger number of patients. But I think this is a fantastic way of keeping patients out of hospital, but giving them the security of tracking their progress and intervening if needed. There are apps to allow interaction with patients with a variety of conditions. So I've been working on esophageal cancer support app with this company called Living With. And you can see that this is just a screenshot from their website that almost any condition you can think of, there is a support app for that. And we worked with them to build an app for patients living with esophageal cancer. And this was really to support the clinical nurse specialists. So these are the nurses who support patients with particular cancers. They can answer questions about, their diet, their pain, fatigue. They're very experienced, but there aren't enough of them. So the question is, could you sort of support the nurse's role by giving an app which allows two-way interaction with the patient. The patient can put in a question. There's a dashboard that opens up on the nurses or the clinician's computer, and they can interact with the patient. And a bit like what I showed you about the previous app, you can have some sort of tracking of symptoms. So the patients can record these sort of things. So if you look at this at the bottom, you can message the patient about their appointments, about their scans. There is also curated information on there. As patient says, well, you know, I've got diarrhea, they can go to the section on their symptoms. It will give them curated information about, you know, what their diarrhea might be due to, what dietary changes they can make. There are outcome measures that we can track on there, the weight of the patient, their fatigue, etc. And, you know, setting goals. So this is a very crowded field, and it is developing all the time. And I think this is definitely going to expand. So wearables and patient support and interaction. And finally, tissue engineering. And this is really a very, very exciting area. So a lot of this has already happened. So we've got basically stem cell technology to try and repair tissues. To you know, it's been done in cartilage, you know, for that sort of injury. My area, as I've said, is the esophagus, and the holy grail for us is, can you replace the esophagus? Now, the truth of the matter is we do replace the esophagus. We replace the esophagus by replacing it with stomach. If you replace this esophagus with stomach, you lose the stomach. So you lose the capacity there. We can replace the esophagus with a colon. The trouble is the colon is a very poor substitute for the esophagus. It's baggy. It hasn't got the same peristalsis, the same way that it can propel food. And my colleague Paolo Di Coppi, who is a professor of pediatric surgery at Great Ormond Street, has been working on this, and he has been pioneering this technology for use in children who are born with something called esophageal atresia. So they have an esophagus that is disconnected. It might be absent, or part of it might be absent. And really, at the moment, we don't have very, very good ways of restoring these children to normal life. Yes, we've got, you know, a stomach pull-up and colon transplants we can do, but people have been working on basically producing a working esophagus by having a shell, that a scaffold, as you can see, which will be in that apparatus there. And this is work that's been done with the Crick Institute. And then you put stem cells on that. But not only do you can you produce an esophagus which is just unicellular, because the esophagus has many layers. You can actually produce different layers of cells. So you can put in the muscle cells of the esophagus as well. The great barrier at the moment is that you can't put nerve cells in there that actually work, and that is, of course, an important part of it. But this work is happening, and the animal studies are progressing. And on the right there, you can see esophagus that's been grown on a scaffold, and you can see it in the cross section on the right there. So watch this space. So Paolo is sort of leading these studies, and I think permission has been granted to possibly use this technology very soon in children born with atresia, esophageal atresia. Neil, that's 15 minutes up. It's really just a panoramic view of a few things that I had in my mind. I thought,
yes,
plenty of time for interaction. Thank you.
No, absolutely. No, fantastic. Thanks very much, Bob. I'm going to start actually with a few of the questions. So you mentioned at the start of the talk that your vision long term for the esophagus is to focus more on imaging. And I've done a bit of work in Barrett's esophagus, not much, obviously not to your level. But is that because, you know, there's all the evidence out there that pathologists often disagree, that there's a big inter-observer disagreement, are you trying to bypass the pathologists and say we can make decisions in real time based purely on imaging, or would you do that hand in hand with pathologists next to you, for example?
Yeah, it's a very good question, and you know, this comes up, as I'm sure you have thought of and may even have come up in the talks that you've had already. You know, there's a profusion of using AI in interpretation of radiology images, for instance. Now, what we're saying for the pathology of the esophagus is, first of all, pathologists are in short supply. It just is a fact of life at the moment. Whether it's the training numbers, we don't know. But of course, pathologists are used in every area of medical practice, from cancer to benign disease, etc. So that's the one thing. We've got a supply and demand situation. So can we augment that? The second thing is a bit like a radiologist looking at thousands of X-rays. You know, 88 X-rays that they look at in a row have got nothing of significance, but the others will. But so there is the drudgery side of it, and then you got the possibility of missing something because you're just looking at one section after another. So the beauty of this is that because this can be digitized, is that you could have a situation where, if we get the right sort of markers, that we can distinguish a deposit of tumor in that sort of shades of gray that I showed you, that the images are producing at the moment, we could have a situation where you could have an AI system which basically classifies the pathology into 3 boxes. One is normal, the other one is abnormal, the second is equivocal. So you could have a situation where it is much better efficient use of the pathologist's time when they are focusing on those cases that the technology sort of puts in the middle box of not sure. Obviously, you would have to do sort of auditing of those other 2 binary boxes as well to make sure that it was getting it right. But, you know, this is what's happening in radiology in many areas. And there is a huge piece of work that's been done on interpretation of CT scans of the brain, you know, matching very experienced neuroradiologists with an AI system, and finding that the AI systems are getting better and better. So I don't see the pathologists being redundant in this. I see better use of the pathologist's time. And the other exciting thing is that sort of 3D imaging, because, you know, when we have to go back, when the pathologist has to go back to look at the histology, they have to go back to the specimen and take more sections. Well, here, you essentially, if you got a machine with terabytes of space, and this is one of the things, by the way, in the technology, I'm told it's a problem. When you got a volume of esophagus that you are scanning, you really need a huge amount of storage capacity to keep all that data. But if you get that information, if you get that data, I can go back and say, look, there is a particular section I want to drill down at. You don't need that specimen anymore. You just need the data relating to the specimen.
Absolutely, fantastic. There's a question from a medical student in Dubai, Faisal Nawaz. He says, lovely presentation. I'm keen to know how important it was to acquire the right collaborations for such novel projects. What challenges do you see in bringing forward surgical innovation in today's time?
Yeah,
so a double question.
Well, I mean, I think they are linked, and it's a fantastic question, because I think this is the whole point of you putting this program together, Neil, because the first thing is that we need to be able to talk to each other. So, you know, I did not know the existence of this X-ray machine, for instance. So there I was in my silo as a surgeon, taking out esophaguses and sending them to the pathologist. And there was, you know, Professor Olivo working in his department, producing a machine, which, by the way, that machine is used for all sorts of commercial sort of things. It's used for checking pipelines and fractures in pipelines, and in security, in doing body searches and things like that. And so they've got this application there, and unless those two minds meet, that universities and have fora where people can meet and say, look, I've got an idea. Could it be that I don't yet know one of the applications that might be out there that I have not thought of? So I think on that front of how easy or difficult it is to find collaboration, it was certainly difficult, I think, maybe 10 or 15 years ago, but I think the world is much more connected now, as we can tell now, because anybody can join what you have put up there. I think the translation is the more difficult thing, I think, is to put these concepts into practice, because obviously therein lies the problem, because you can have fantastic ideas, but if you don't have the traction with the funding, etc., then it can be a problem. So we're lucky that we got the funding to do this project, and we've got the right sort of networks within UCLH being connected to UCL for us to be able to do translational research. But I would say that that is the more difficult aspect, is to translate the science into something that works clinically.
Fantastic. Yeah, makes perfect sense. We have another question around, probably focused on the medical curriculum. So medical students ask, pathologists mostly are interested in 2D specimens. Medical students, as they become doctors, they're learning concepts of diseases on pathology in 2D. With the help of micro-CT technology, do you think 3D pathology patterns may be incorporated into the curriculum?
Yeah, I think that'll happen. But you see, I don't. Was the medical student asking that question, or?
I think so. Yeah.
Well, the reason I ask is this: that my experience of having worked with universities and the examination departments is that it's incredibly difficult to change curricula. They have to go through all sorts of layers of approval, and I'll tell you why. Because people who sort of are in charge of university or medical curricula get lobbied by pathologists. We want more pathology. Rheumatologists, we want more rheumatology. Psychiatrists, etc. So I think it becomes very difficult for them to sort of change curriculum radically quickly, but I have no doubt that this will come into practice. Now, let me give you another example of this 3D pathology, and that is to do with imaging of endoscopy. So, Neil, you mentioned about your interest in Barrett's, for instance, and as you know, the Barrett's story is that you can look down an endoscope and an experienced endoscopist will say, I think that looks like Barrett's. But of course, as you know, you got to do biopsies for the pathologist to look at. And as you know, sometimes you need two pathologists to sort of decide whether this is high-grade dysplasia, low-grade dysplasia or not. Now, the Japanese, who have done a huge amount of research in cancer of the stomach and the esophagus because they got a high incidence and prevalence of these conditions there. And they also have, Nikon and Fuji that are based in Japan. They have produced endoscopes now. So you just put down the endoscope and you can increase the magnification to such a level that it gives you a 3D picture of the mucosa. You can see the capillary loops underneath. And they've started to publish textbooks of endoscopic pathology so that you can actually determine from the pit patterns of the mucosa whether this is a benign polyp, it's a polyp at all, etc. So not even having to do the biopsy. So I think that's right. It is something that if we are at that stage in the future that we are having to train doctors to do that sort of examination, then it is, if you work it backwards, you have to put that into the curriculum to say that that is actually possible. So in other words, maybe in 30 years time, it won't be that if you suspect somebody's got Barrett's, you do an endoscopy, you see what the mucosa is like, you biopsy it, you send it off. It'll be, you use this high-end, high-resolution endoscope, giving you 3D vision, and you put it down there, and you make the diagnosis of Barrett's there and then.
Yeah, real-time diagnosis.
Real-time diagnosis.
There was a lot of, well, speaking about the Japanese, yeah, and I agree with you. They're trained to just look at something and they get it. There's no need to take that biopsy.
Yes.
And I think there's lots of reasons why they're trained like that. I think, efficiency for one, probably, like you say, the pathology, gastric, definitely in Japan is higher than say here.
Yeah.
And also esophagus as well is probably more in the West, but I think that's changing, isn't it? With
it is changing. It is. I mean, we hardly see gastric cancer now in the West, but esophageal cancer, although it's plateaued a little bit, is still rising. And sadly, the UK has got worse figures than Europe for some reason for the incidence of esophageal cancer.
I think, like you say, it's to do with the training at endoscopy. Are we looking in the stomach in more detail, or are we just looking for big things? And if there's nothing big, then we finish the procedure.
Well, I'll give you an example.
Sure. Sorry.
The average time for doing an endoscopy, when you don't see anything particular, is about 6 minutes in the UK. So you just go down, you have a look, you come out. In Japan, the endoscopy is done over at least 10 minutes. So they look at every little thing. They suck out the saliva. They wash the food away. They put in, indocyanine dye spray to see whether there is a slight area where the uptake of that dye is different, and they biopsy it. When we biopsy here for Barrett's, we might take 6 biopsies. They will take 4 or 5 biopsies around the circumference of the esophagus, every centimeter for the length of the Barrett's. So, it's a much more detailed and laborious process. But of course, they are also designing the technology to make it less laborious, but more efficient.
Yeah, they're investing the time, which I think I often hear in an NHS system that often, there's so much pressure that because of the backlog of procedures, it's better just to do the procedure, spend that 6 minutes, and then on to the next one, which is not necessarily the best thing long term.
No, you're right. You're right.
I wanted to ask you, if I may, the slide that you showed, I thought was fascinating. So, like you say, the interplay between medicine and engineering, a lot of movement with the wearable devices.
Yeah.
And then you mentioned one on sepsis. Yeah, exactly. So it's interesting that even though things were kind of going wrong and was detected, that say patients weren't necessarily.
Why there was no intervention?
Is that a slide looking, just from my understanding, is that looking at patients when you say sign of trouble?
Yeah.
They then being triggered to say, okay, present yourself to A&E because you're.
No. Interestingly, yeah. So Neil, it's a very good question, because this is exactly what anybody looking at that slide without the text says. Well, why were we waiting this long to go to A&E? So when this study, the pilot, was set up, it was set up in a way that no intervention would be made on the basis of the wearable.
Oh, fine.
That sounds the ethics sound funny, doesn't it? That you would say that you're watching somebody having a tachycardia, but the point was to study when the patient actually comes in to A&E with whatever symptoms they thought needed to be dealt with in the A&E. Then the next iteration of this study is exactly that. You don't wait till there, you interact here.
Sure.
So this was an observational study.
Because my head was just trying to, because often these concepts, you can parallel it to something else. So I'm still training, almost finished, but we had to spend time in the liver transplant unit for the training. And obviously alcohol is a big issue So a lot of decompensated alcoholic liver disease. And they come in, they're jaundiced, you take a blood sample, you know their bilirubin is sky high, ammonia's off. But whether there was a way that when they're at home, if they had that capability, or if there was a system in place that was able to detect in real time at home, if they were drinking, that, okay, your ammonia level is creeping up. If you keep drinking, you're going to become encephalopathic. So then, okay, we can't stop you from drinking, because I think governments have tried that. It's never going to work. But in terms of the wearable area, I think that's really interesting, because when they come into hospital, it's a lost cause most of the time.
Well, I think you're absolutely right. So one of the as I said, this is a crowded space. Some other app manufacturer contacted me recently and said, would we be interested in their app, which monitors people who are having chemotherapy. So a bit like the example that you gave, Neil, about somebody who is just about to fall off the cliff with decompensation of their liver problem. As you know, one of the side effects of chemotherapy can be leukopenia, which can occur very quickly. And of course, they're then prone to sepsis. And so this app was designed to monitor that aspect, of side effect of chemotherapy, and so that the patient can come into the center very quickly and have the right treatment in terms of stimulating. They can be given sort of granulocyte colony stimulating drug, which brings their platelets up and their neutrophils up. So yeah, I think this is a fascinating area. And of course, as you know, one of the wearables which is now extremely successful is the continuous sort of monitoring of blood sugar. So they've revolutionized the lives of children, because obviously a child, child can't interact and grow normally and play normally if you continuously, taking them into a place to take finger pricks and, deciding what their blood sugar is. And of course, those sort of diabetic monitors are now linked with devices that will give insulin as well appropriately. And the other thing is that they can be monitored remotely. So, the mother or, the specialist nurse gets a signal that child A is, in this state, or their blood sugar is doing this, that, and the other. I think there's a tremendous potential in this area, in all sorts of medical conditions, to optimize and, genuinely improve the quality of life, because you don't have to be going into a GP surgery. You are being monitored remotely in an expert way.
Absolutely. No, I agree. And I think, like you say, you want to get to the patient or the individual before, it's way too late. And then we're trying to, reverse everything whilst they come into the hospital.
Exactly.
I do have another question, lots of questions, but I know you're very busy. So the element of tissue engineering, very exciting. Again, your focus is the esophagus, so we give them PPIs, or you can burn the tissue. And then the very good endoscopists, which I'm not, they can resect. So do you think in the future that there would be an ability where you can see something that's inflamed in the esophagus or the stomach, and then at endoscopy you're spraying, kind of healing factors or something like this, which is kind of linked to the tissue engineering? So you're directly applying a plaster, if you will.
Yeah. No, this is absolutely. So this has already been tried. So people have used these sort of, I'll give you an example. We sort of, in my sort of complex clinical practice, dealt with tracheoesophageal fistulae and things like that. And as you know, that's a sort of a connection between the airways and the esophagus, very, very difficult to repair. big operation requiring access to the chest and the airways and everything that goes with that. And, people have tried for many, many years innovative techniques. Can we plug that? Can we put clips on them? Can we have an endoscopic device, which will stitch that, etc., without having to open people's chests, etc.? So people have now started to, and these have been trialed, to produce a sort of tissue engineered plug which has stem cells on that, for instance, so that you can plug the gap and then enable normal healing and normal epithelialization with stem cells in the same way as orthopedic surgeons. I think your example is equivalent to an orthopedic surgeon doing an arthroscopy, finding that, a chunk of the cartilage is missing and putting in a little graft of scaffold onto that missing piece with stem cells for the cartilage to regrow. So I think it is only the equivalent of that in the esophagus or other places, other parts of the body. And so I mentioned Paolo Di Coppi with the esophagus. So one of the other things that his group are doing, and that's a little bit of a few years ahead, is they're looking at transplant of organs. So as you know, transplant, there are lots more people on the transplant list than organs that are available, and sadly many people die whilst they're waiting for organs. There aren't enough there. So one of the things that their group has been looking at is for kidney transplants, taking a kidney from one animal, for instance, and completely taking away all organic material from that and just leaving the sort of collagenous structure, non-allergenic collagenous structure of the kidney. So if you think about it, it's the equivalent would be to have the sort of, we've all seen sort of the leaf structure of the stems when the autumn has sort of completely taken away the greenness and then everything has melted, but you just got a structure of the leaf. And then you seed that with the stem cells of another animal so that you produce something that is allogeneic to that animal in terms of transplanting that. But they have had some sort of success in that. So tissue engineering will also be something that is used to grow organs, not just repair them for transplantation, and maybe changing them from one species by taking the species out of that organ and seeding it with the species, if you like that you're going to transplant that into.
It's incredible stuff, and I think what's more incredible is that you're still so dedicated and passionate, which is it's really remarkable. So thanks very much for your time.
Thank you.
We're going to take a break now, everyone. So we have Dr. Helen Meese coming up at 5 o'clock. So like I always say, have something to drink, stretch your legs. We don't want to sit all day. And so we'll be back at 5.
Thanks, Neil. I'm going to log off now, but I should have said this earlier on, some of these slides I've shown are sort of pre-publication, so please don't disseminate them.
No, we won't. We will be discussing how to edit this in the best way, so don't worry.
Thank you.
Take it. Bye.
Bye-bye.
Okay, hi everyone. So we're just getting ready for our final speaker. I'll just bring her into the session. Hello, Helen, can you hear me?
Hi,
Perfect.
Neil.
Hi, how are you?
I'm good, thanks. How are
That's
you?
awesome. What is, we've got a great device here.
I do a lot of audio stuff and a lot of podcasting. So yeah, this is my welcome to my studio. It's a little dark in here, but
No, it's really cool. I'm very jealous. How's your day been so far?
it's been pretty good, thank you. Yeah, it's been relatively busy today. Yeah.
I'm sure. I'll just give it a couple of minutes and then we can start at 5.
No worries.
Okay, we can kick off, I think. Okay, hi everyone. So this is the Clinician Engineer Hub virtual conference over 3 days. As I always keep saying, we've brought to you the best speakers across academia and industry on an international scale, and that the benefit of this is that it's completely free of charge. So we have our speaker closing the first session, Dr. Helen Meese. Dr. Helen Meese has multiple years of experience in academia, industry, and engineering, I think well over 20 from her bio. Her bio states lots of great things in terms of being the CEO and founder of the Care Machine, massive amounts of experience at IMechE, and I think more recently, writing and compiling the report looking at why the NHS, the National Healthcare System, for those of you not in the UK, why they actually need to understand this interplay better with healthcare and engineering. So I'm going to pass it over now to Helen and go for it. Yeah, we're all ears. Very excited.
Excellent. Thank you ever so much. So I'm going to share my screen.
Mm-hmm.
Hopefully you will be able to see that okay.
Yeah, we can see it perfectly. Thanks, Helen. Thanks.
Excellent. And what I'm going to do as well is I'm going to turn my video off so that you don't get distracted by my face and you can listen and really get into the content of what I'm talking about today. So, okay. Well, good afternoon, everyone. I'm Dr. Helen Meese, as Neil has introduced me. I'm CEO of the Care Machine, and welcome to my presentation, which is entitled Engineering Healthcare Solutions, the Future of Engineers and Technology in the NHS. So please feel free to use the Twitter tags in the bottom corner during the talk, and also the QR code, which is also on the last slide as well, to visit our website. And I'm more than happy to answer lots of questions at the end. The Care Machine is dedicated to ensuring that technology is at the forefront of our healthcare service provision. And I believe innovative medical technology should be available to and improve the lives of everyone. And the pandemic really has shown that really is true in all cases. We are on a mission to support entrepreneurs in the medical technology, healthcare and associated sectors by combining our unique engineering and science expertise to help produce innovative technology solutions. And we do that by facilitating and participating in our clients' ideas. And through this, we achieve our vision, and we are committed to ensure that technology enhances the human touch in healthcare, not takes it away. Now, I personally have over 20 years experience in industry, academia and public affairs, having started my career in the defence industry. Today, I am Vice Chair of the Biomedical Engineering Division at the Institution of Mechanical Engineers. and I'm also a member of the Healthcare Policy Panel at the Royal Academy of Engineering. And it's as the Biomedical Engineering Division Vice Chair that I present this talk today, focusing on two recent reports published by the IMechE in July this year, of which I was lead author. I must first really, though, acknowledge that these documents are the combined efforts of a number of IMechE members and policy staff, with input from experts across the institution to ensure that we've reflected the views of our engineering colleagues across the healthcare and medtech sectors. So let's get started. Like doctors, chartered professional engineers and technicians have a statement of ethical principles in which we aspire to do no harm by ensuring that we do not compromise where society's welfare and safety is concerned, and that we listen and work with society to create transformative technology for the right reasons. It follows then that care for society forms a fundamental imperative for engineers in healthcare and is at the forefront of all we do. Despite incredible progress, healthcare services across the globe will continue to suffer from mounting pressures, driven by an increasingly aging population with expectations of continuing health and independence into old age, lifestyle conditions, and also the impact of poverty in poorer countries. At the same time, we are witnessing a rapid expansion in healthcare coverage in developing nations and a medical technology revolution driven by health data analytics and the digitization of patient care pathways that has enabled previously intractable conditions to be treated. Global crises such as the pandemic we are in right now have highlighted that 21st century medicine can only be delivered with a significant amount of technology input, and that care at home is just as important as care in hospitals. The rapid change in healthcare provision will require comprehensive strategies from governments across the world, regulatory authorities and healthcare providers to allow for timely development, evaluation, implementation and regulation of all of this technology. Today's presentation will focus on two areas highlighted in the IMechE reports. The first being the value of engineering in healthcare provision, why more engineers will be needed in the future, and what influence they could affect within the healthcare service. The second being, what can this engineering workforce do to address the slow pace of adoption of technologies, and how might our healthcare providers benefit from their professional understanding of technology in a rapidly changing environment? Well, before we get into the discussion of these particular points, let's first look at the healthcare services. And for the purposes of this presentation, I'm going to focus on the UK NHS. But I do believe much of what is presented here could be applicable to many services across the globe. For those of you listening today who are unfamiliar with the workings of the NHS, it is a relatively unique system which is free at the point of care for all. It has 1.5 million employees, making it the biggest employer in the UK. And just for me to put that into context, the British Army only has 125,000 soldiers, and Tata Steel, one of the world's biggest manufacturers, only has 80,500 employees globally. So that just gives you an idea of the size and scale. The NHS sees about 1 million people every 24 hours under normal circumstances, of course. And the combined health estates stretch to 66 square kilometres. That's about 25 square miles. For those of you in the UK, it's about the size of the city of Lincoln. It's recognised the world over as one of the most effective and efficient care systems. Yet, like any healthcare system, it needs constant improvement and development to ensure it provides the best care to patients. And this costs money, lots of money. And presently, the NHS consumes about £135 billion a year. That's obviously a figure from pre-COVID. But if you look at this in terms of the per person amount, it's about £2,300 per person per year, which doesn't seem all that much, really. But this cost to taxpayers is only going to increase over the coming years as our population grows and ages, and thus treatments for long-term illnesses escalate. So to put that into context, if we compare that to global health, almost half of the world's healthcare expenditure goes on preventable illnesses, such as respiratory and cardiovascular disease, often brought on by lifestyle or environmental conditions, and also cancers. On top of this, we are seeing a significant fall in the number of frontline healthcare workers, mainly doctors and nurses, particularly in developing countries. Supporting this advancement in care provision, however, is the medtech industry itself. It's already a £356 billion market, which is set to surpass pharma within the next 5 years in terms of size and scale. There are over 500,000 CE mark products available in the EU for hospitals to purchase, and most UK hospitals have somewhere between 20 to 50,000 devices in service within their trusts. The unprecedented swell in healthcare devices, apps, and services across the globe, certainly as a result of COVID, clearly shows how ubiquitous technology is, and it's only set to rise. So with this in mind, what changes can the engineering workforce affect within healthcare? And what can we do as a community to ensure that we have greater involvement in the patient care pathway? And what will happen if we don't? Well, as I've already stated, technology in healthcare is everywhere. Pre-COVID, it's safe to say the existing healthcare workforce had the capability of taking on greater responsibilities for technology adoption and development and for significant engagement in patient care. However, if we continue on a path of high technological demand based on new and uncharted disease conditions, and with yet fewer and fewer engineers particularly available or entering the field with the clinical expertise to design, manufacture, and implement that technology, we will reach a point where we will be unable to meet patient need. So let's take a moment to look at the kind of roles engineers do in healthcare at the moment. It's often hard to spot a clinical engineer in the NHS, yet many do actually work at the patient bedside. These engineers develop devices which diagnose, treat, and rehabilitate patients. They create technology which helps measure, model, and simulate human physiology and anatomy, which enables doctors to make ever more precise diagnoses. They are the pioneers of artificial joints and organs and robotic surgery equipment, which has advanced minimally invasive procedures over the last few years. And they have explored the growing area of monitoring personalized health through mobile communications, allowing patients to be in contact with their doctors 24/7. And they also ensure the vital supply of gas and air arrives in theatre when the surgeon needs it. From Sir Godfrey Hounsfield to Sir Peter Mansfield, the early pioneers of CT and MRI scanning, to the life-saving cerebral blood flow techniques conceived by Professor David Delpy, engineers have been changing the way healthcare is provided for decades. More recently, if you've seen the news, Professor Chris Tomaszou, who has received over 25 awards for his pioneering advances in DNA sensing and vital signs monitoring, has been actively developing a new technique for the rapid detection of COVID-19. His new portable test, which combines biochemistry, microfluidics, electronic circuits, and miniaturized systems based on smartphone technology, can give a test result in just 90 minutes, which is quite incredible considering the time of development that's taken. And these are just some of a few of the hidden faces of engineers in healthcare, both on the front line and in the medtech industry. Now, the reports suggests that healthcare engineers should become even more involved in the direct delivery of care to patients, and an appropriately trained workforce is key to ensuring the delivery of safe, affordable care on a global scale. Failure to act on these requirements will increase the risk of patients' health in the long term. So it follows then that healthcare engineers should have professional parity with their clinical counterparts, particularly in the NHS. That's quite an outlandish thing to say, I suppose, and I'd be interested to hear your thoughts. But to achieve this, the healthcare engineering workforce is going to need to take on new skills and responsibilities to adapt to the changing healthcare landscape. This would provide a much broader end-to-end systems perspective to care, which traverses the more vertical, specialised expertise of typical clinical models. The Institution of Mechanical Engineers is committed to ensuring that the views of engineers are well represented in the public domain, and that its members embody the institution's vision to improve the world through engineering. So in response to international healthcare challenges, the institution defined a professional commitment for healthcare engineers. This commitment urges them to ensure that every hospital has professionally recognised engineers and technicians with consistent qualifications, levels of authority, and decision-making abilities. To achieve this, we defined a professional commitment statement, and this is taken exactly from the report, which recognises the healthcare engineers' commitment to do no harm, but also recognises that healthcare professionals trained in both clinical and technical competencies will be able to ensure patient safety through implementation of quality processes, provide value for money through benchmarking, and ensure faster access to medical technology through trialling and product development. But to achieve this goal of recognition, two key roles need to be created. Every hospital should have a chief healthcare engineer, a position of professional parity with roles such as the head of surgery, chief nurse, or even chief pharmacist. These engineers would have consistent qualifications, level of authorities, decision-making abilities, and connectivity with other hospitals. This would not only promote best practice in procurement, maintenance, and use of medical equipment, but it would increase the opportunity for cost savings across the healthcare service. In many hospitals, the concept of chief healthcare engineer may already exist, albeit under different titles, departments, and levels of authority. But the cost of creating this board level position would be recovered from the long-term benefits to patients, services, budgets, and healthcare providers themselves. The growing desire to move long-term care and treatment out of the clinical setting and is creating particularly new dilemmas for an already overwhelmed social care system within the UK. And steps need to be taken to build on the well-proven techniques of rehabilitation and assistive technology engineering to create a patient care pathway at home. Patient enablement engineers and technicians would work exclusively in the space between acute care and social care with their clinical colleagues. They would not only require the full remit of engineering qualifications and skills, but in-depth clinical and social care knowledge, as well as management and customer services experience. Professional engineering institutions really do need to work with the healthcare providers and healthcare engineering leadership organisations such as the Academy for Healthcare Science and the National School for Healthcare Science in the UK to create new training programmes in areas such as process flow, procurement and purchasing, and strategic planning to support engineers working in the social care area. And such harmonised approaches across the healthcare engineering profession would really raise the profile and level of importance of engineering in both clinical and medtech manufacturing, aiding recruitment and retention of engineers across the sector. Through this, engineering will become much more visible, providing direct services to patients and ensuring that long-term strategies such as the NHS's 10-year plan are fully implemented. Now, this new decade is expected to be nothing short of amazing when it comes to medical devices. And I'm sure you've heard from a lot of the speakers today who've talked about some incredible developments that are going on. And certainly the current pandemic has had an effect on that. But we are seeing a speed and scale to medtech development, which we've not seen in the past. We will see an increase in multidisciplinary technology entering the sector, including more personalized sensing and diagnostics and miniaturized robotics, which will certainly herald a new wave in surgical innovation. But in the case of the UK, the structure of the NHS has yielded significant challenges when it comes to the development and delivery of technology to the front line. With its mix of centralized authority and regional self-governance, the route to technology adoption within the NHS can, and I'm sure several of you will agree, be an arduous one. The NHS is often seen as being resistant to innovation, which does not deliver an immediate tangible benefit to patients or to cost savings. And this can stifle both medium and long-term innovation strategies. The adoption of technology will be affected even further as the requirement becomes more complex and multidisciplinary, with greater interconnectivity, integration, interoperability, and interdependence. Now, in the UK, the NHS has created a long-term implementation program framework to improve overall care for patients, which includes a drive to digitally enable service provision over the next 10 years. Alongside this, the government has oh, sorry, I've jumped one. The government has launched its Tech Vision Plan in 2018, outlining the introduction of technical standards that digital services and IT systems in the NHS have to meet. While this digital transformation will address some of the connectivity and data management issues facing the UK's health service, it really does fall short in looking at ways to improve medical device and equipment procurement and adoption in the long term. Technology innovation in the NHS is often a very time-consuming and costly process, even if it's an innovation which is relatively simple. And uptake of technology in the UK has been slow, and this has often been due to isolated approaches to procurement and inconsistent distribution of innovation within and between NHS trusts. These entrenched approaches to innovation have often resulted in unsuitable or poorly specified devices, and in some cases, technology is being incorrectly used or disposed of because it fails to meet the needs of the clinicians and patients. It's likely that if steps are not taken now to create sustainable strategies for a complete life cycle technology adoption process and greater engagement with engineering specialists who oversee it, the NHS may be unable to propagate a coherent and cost-effective approach to patient care in the future. That's kind of doom and gloom, I suppose. But more significantly, there remains a mismatch in the UK's health service between the money invested in developing technology versus the investment to adopt and spread technology at scale. In a King's Fund report from 2018, the NHS was shown to set aside less than 0.1%, that's about £50 million, of available resources between 2013 and 2018 for the adoption and spread of technology, compared to the 1.2 billion it spent on investing in R&D. And according to the World Health Organization, progress in adopting technology varies wildly from country to country. However, successful national technology adoption often goes beyond patient need and funding and requires the engagement of all healthcare stakeholders to deliver successful uptake. So the role of GPs, social services, pharmacists, and even family carers must not be overlooked when when we consider how technology should be adopted. Empowering patients to play a more active role in their own care, for example, through remote health monitoring or regular local testing at GP surgeries, could drive greater acceptance, compliance, and adoption of technology. So here lies an opportunity to create a national strategy to address care within the community based on regional need, which could yield significant technology uptake. This could be managed through a nationwide network of engineers dedicated to patient enablement, providing patients with access to the right devices when they need them. In order to address the growing demand for more healthcare and the technology to support patient care, an additional workforce is needed that can advocate and implement technology at speed and scale that the NHS can feel more comfortable with. To achieve the level of technology adoption in the coming decades, engineers will have to have a greater presence across the NHS to interact with and interpret clinicians' needs, understand the patient care pathway, and be able to validate the devices in question. Engineers across the healthcare sector must therefore be empowered to take greater ownership of the procurement and implementation processes for technology across the hospital networks. This would include being directly involved in the design, selection, purchasing, maintenance, and decommissioning of medical equipment, adapting it to local environments to ensure it is fit for purpose, sustainable, and cost effective. Now, within the report, we stated that to deliver safe and affordable care in a fast-changing healthcare environment, government and healthcare providers need to look at implementing a national complete lifecycle technology adoption strategy. This approach would ensure equipment is correctly specified so it's fit for purpose and delivers the expected patient benefits within budget, as well as being managed sustainably through to the end of its life and disposal. Again, healthcare engineers are already well placed to carry out this kind of role, but the benefit of their involvement will only be realised if a more consistent and coherent approach to their work and skills is implemented. The IMechE has identified 2 opportunities which could offer the most widespread engagement with and uptake of technology across the NHS network, and would also significantly benefit from complete lifecycle technology strategies. With many countries shifting healthcare focus to prevention rather than just cure, greater pressure is being placed on primary care services. So there is an opportunity to establish national technology procurement and adoption strategies specifically aimed at GP surgeries. With this would enable a much more holistic prevention program to be delivered, really. These kind of advanced GP surgeries would contain in-house early diagnostic technologies for liver, lung, and bowel cancer, for example, as well as advanced blood testing facilities, analysis, and other preventative technologies. And by enabling patients to be tested locally, waiting times and referrals could be reduced to days, not weeks, saving thousands of pounds per patient and billions of pounds in long-term acute care. Additionally, click over. No, it didn't. Apologies. Additionally, remote monitoring is becoming increasingly important for managing the impact of conditions such as obesity and diabetes, age conditions such as Alzheimer's and frailty, as well as end-of-life palliative care. So effective management and support of remote monitoring systems and home diagnostic applications in social care settings will need both technically and clinically trained professionals. Back in 2017, the institution called for the creation of a national remote health monitoring network, and this was reiterated in our 2018 policy on intelligent assistive technologies. A remote healthcare network that would fully integrate acute and social care using existing data analytics, wearables, and sensors could completely revolutionize health provision by easing logistical pressures and supporting care initiatives for patients outside of the hospital environment. A key element of this would be to standardize the remote health monitoring equipment that enables patients' information and care needs to be accessed anywhere in the healthcare network. With the constant turnover of monitoring equipment, a national complete lifecycle technology adoption strategy would ensure that patients have access to and benefit from the latest technologies, which are fit for purpose and tailored to their needs. So, in conclusion, the Institution of Mechanical Engineers is committed to ensuring that performance, safety, and ethical standards are maintained across the profession when creating transformative technologies, such as those used in the healthcare sector. The institution is well placed to inform decision-making at all levels of government and care provision, and this will ensure the highest levels of care and safety for patients, and that where technology and services are being implemented, they are the right tools for the job. In an ever more technical surroundings, we need engineers to ensure our healthcare providers are able to make the best decisions possible regarding medical equipment, which can facilitate early diagnosis and provide better care and more effective treatment for patients. In response to international healthcare challenges, the institution urges all healthcare engineers across the globe to establish and share strategies and best practice processes for life cycle technology adoption through engineering communities and networks. Innovation cannot be spread passively. To ensure active acceptance by patients and practitioners, it requires insight into the application, awareness of the clinical setting, and an in-depth knowledge of the device's function. The realistic and long-term adoption of technology can no longer be the sole responsibility of the clinician, but must include healthcare engineers who are able to implement innovations and adapt them to the local environment, ensuring patients have a safe passage through our healthcare system. Thank you very much.
Awesome. Thank you, Helen. That was really fantastic. I'm going to start off with the questions, if I may. So, from my personal background as a clinician, I fully agree with you that, the NHS definitely requires more engineering influence. And what I find interesting is I work at the Queen Elizabeth in Birmingham, and I see the engineers, and it's exactly as you say, that they're hidden. And I don't really know why that is. It's like we see them fit into our technology based issues. If we have a problem with the fluid pump, or if say the endoscope is not working, or the dialysis machine has decided to not do what it's supposed to do. And I think that's the problem. Like I don't know why that is, because they're clearly there. All the engineering platforms are there, but yet we still are in a very strange mindset that they're just not really present in a way, and I don't really know how to tackle that.
It's a constant conversation that I've been having with the clinical engineering community for many years now, and I'm still talking about the same things I was 5 years ago. So clearly there's been very little change. I think what some of the biggest problems have been that they're seen as a last resort, as it were. They're seen as, oh, they just fix equipment. And so, if a pump goes down, if a life signs monitor fails, so they come, they take it away. You never see them fix the thing or replace it or service it or do all of the maintenance things that you might expect, because it's done usually in the bowels of a hospital somewhere. And I think so. I think it's been a lack of visibility. But I think what has brought them to the fore recently is certainly within the Nightingales, and I've been involved with the Nightingale hospitals, certainly down at London and more recently across the UK, is that those engineers are now right at the bedside, because the speed of care has gone up so quickly that they have to be at the bedside in order to work with the clinician to solve problems then and there. So their presence has become more widespread. But also, I think because we've seen a lot of development around the CPAP and the ventilator challenge and so on, that we've suddenly seen what engineers actually do. And it's not just been within our community, but actually it's been on the news and the TV and all of these things, that suddenly engineers are solving these problems. So I think it's very important for us as a community to not let that go back to us being in the darkened rooms in the basement fixing equipment, but actually to be present and to be active within our communities, and hence the reason for these two reports, really.
Absolutely. And in terms of that position that you mentioned, the chief engineer, and I agree with you, because in every hospital there is a chief pharmacist or there's a chief registrar, and then the concept of the chief engineer. How are you trying, and I'm sure it's a lot of work, how would you try and implement that from, say, the policy document to sort of real life, everyday working?
Well, it has to come from multiple directions. Obviously, as an institution and as lead author of this report, having had those conversations, there are a lot of people who want to make this happen, but it has to be from multiple directions in order for that to happen. I think one particular area, and I think we'll start to see that come through over the next few years, is from the likes of the National School for Healthcare Science, for example, who are doing amazing work at revamping and have done over the last few years, revamping the structure of the training for clinical engineers. And so encouraging those engineers coming through that professional route to aspire to be chief engineer, and to aspire to change the way it's always been done within the hospital, I think is going to be fundamental. So it has to come from the engineers themselves. And I think that drive through the professional development of engineers will be key. But there's also an opportunity, I think, for government to make policy change that could encourage the NHS to raise the profile of those engineers and raise the professional status within the hospitals. We're starting to see the odd, chief engineer coming through, but they're reporting into a scientist or a healthcare scientist or someone who's maybe not in the engineering field. And I think we now need to recognise that engineering needs to be a recognised workforce within the NHS. I think that's very important.
No, that makes complete sense. And I think, it sounds, if you are recruiting a chief engineer, but they get allocated to a scientist, obviously that probably isn't going to match up very well. I wanted to say also, do you think you mentioned that the NHS is a massive organization, 100% agree with you. And I think the NHS does a lot of fantastic things, but because it's so big, any change can be quite disruptive and probably, I think, have a negative impact or a negative perception. Now, so my experience as someone training in clinical medicine is that as I was going through my training, we were trained around, okay, if you need to be a good physician or a surgeon, you need to know how to talk to patients or take a history, physically examine patients, and then you'll put in a diagnosis and management strategy. But in reality, what we were seeing, even though that was the medical school way of training, and that's what you needed to do to pass exams, when you then exit, every time I've been on call, I've always relied on an engineering solution. So if someone's got a kidney problem, I can give them fluids, but we're going to dialyze them, like you say, with COVID-19, breathing issues, ventilation, endoscopy we've heard about earlier today. My experience has been that I think the higher ups get a bit nervous around all this engineering technology. So do you think it's just a matter of waiting for them to step down, or do you think we have the power to, persuade them that this should be something that you should accept and take on board?
It is difficult to make change, and it's always going to be sort of evolving rather than kind of some major revolution that occurs, really. I think, yes, I think the younger cohorts of clinicians need to be driving that change, most definitely. I think it is very difficult when you're in a senior position. You're responsible for the cash. You've got to make balanced sensible decisions. And I've been a trustee of a charity. I know exactly those difficult choices that you have to make. But I think it's about encouraging the senior management to be much more open-minded to the changes in practice, I think, and the changes in culture, I think, amongst the clinical and technical communities. Their attitude towards science and technology, I think, has changed significantly to certainly when I trained to be an engineer, and even just in my working career, I've seen significant changes. So I think it has to come from the bottom up, most definitely. And it will take time to make those changes happen. But when we see significant impacts such as COVID and things like that, I think that, again, brings it to the fore and makes the senior managers really think differently about how they're going to problem solve. And also, we're all well aware of what was stated in the Topol report. I think when you have somebody at that sort of level making recommendation to government to say clinicians need to be taught in technical subjects, then that is exactly what we need to be doing. And that will, so the next generation after you and after them will be trained clinicians, but will have done some medtech, study as part of their training. And that will be great.
Yeah, no, exactly. And I think, like you say, the culture of innovation, if you look at the US, obviously they're always mixing it up, changing things. And I think sometimes it is difficult here, but actually, if we talk about it more and we do these kind of webinars and interactions, there's no reason why the UK can't lead the way on this.
Yeah, absolutely. I think it's important, as I mentioned in the presentation, it's important for us as a community to come together. Excuse me. And it's important for us to share best practice. The organizations, for example, like the Q community within the Health Foundation, I found that to be an incredibly useful community to join, because it's given me a chance to network and discuss subjects around quality and quality improvement processes that I wouldn't have had the opportunity to do before. So I think as engineers and clinicians, I think we need to be looking for those opportunities through this kind of event over the next few days, and more importantly, joining organisations where we can create those networks and build that little black book of names that we can call on when we've got a problem, and we need that multidisciplinary team to come together to solve it. And it may be that there's a doctor somewhere, I don't know, maybe in Newcastle, who could really help you solve a problem, when you're in the midst of diagnosing a patient. So we've really got to work at that, but that's got to come from us as a community.
And just a final question, and then I'll let you have your evening off.
It's all right.
Do you think, obviously you've got massive amounts of experience in this. You've led the report, you've been pushing for this for many, many years. Have you experienced, you mentioned maybe resistance from government, but how about regulation within clinical medicine? So we are all heavily regulated, and often I think sometimes that puts us off for trying to do something which is slightly different. Do you think that conversation needs to be had with regulators as well? Or?
Yes, absolutely. I mean, I always give the example that, if you think about medical devices and medical technology, it's as heavily regulated as it is to build an aircraft carrier, and it can take just as long in some cases. So we, as I said at the beginning, we have an ethical responsibility to do no harm as engineers, and you as clinicians also have that. So it will take time to make changes to the regulatory requirements that are placed on us as professionals. That's not something that's going to happen overnight, but the only way to change regulation is to push the boundaries. And so we need to ensure that anything that we do in terms of developing best practice needs to be presented in a way through policy to government, through consultation, and that's something that we as engineers do an awful lot of. When government asks for consultation on a subject, we provide them with our views, and often that can make significant change to policy and to regulation. So I think, again, there's an opportunity for us as a community to be setting the conversation, setting the discussion points, and raising that and bringing it together in a coherent way that we can present to the regulatory authorities to help them develop these documents in the future and help us develop as professionals.
Perfect. Well, thanks again for your time. That was really great. Very detailed. A question has come in, if I may.
Yeah, absolutely.
Okay, so from Dubai, we've got a question from Faisal. Do you see a need for change in the curriculum for both engineering and medicine in becoming more hybrid in future practice?
Absolutely. I mean, we're starting to see that in the UK through the biomedical engineering degree courses, of which in just in the last few years, I think there's now like 100 and something degree courses in biomedical engineering purely in the UK, which just didn't exist when I was applying for my engineering degree. So I think we are going to start to see, we're certainly seeing in the engineering sector that crossover between medicine and engineering, with engineering being the predominant part of that degree, if you like, the major, and then the minor is clinical. I think we need to see that in the clinical sector. So a medical major with an additional sort of training in engineering, technology and science specialisms that would sit very well alongside the clinical engineering curriculum. And I think we will start to see hybrids of clinicians being, trained in engineering and sitting in, fluid mechanics lectures or whatever, because you understand fluid mechanics as well as we do, just in different ways. But the principles are the same. The mathematics is the same, and so on. So I think there is some great opportunities for people in the future to be, medical engineers. That would be amazing.
You're definitely leading the way, so it's awesome to have you here for our...
I'm very passionate, as you can tell.
Okay, well, thanks everyone. So that's the final talk for today's conference. We'll be back again tomorrow, starting at 1 o'clock UK GMT. As you can see, we have all the best speakers, I would say, from academia and industry. So I'm going to close there. And if anyone has any questions, you can contact us at the Clinician Engineer Hub website. We have our email and social media details there. All the best. Thank you.
Thanks, everybody. Bye-bye.
Bye.